So, hello and welcome to the 8th of the series of Advanced in Silico Drug Design workshops. We are offering you this workshop both on site and online via the YouTube channel, and we are offering it with the help of these 25 people who help to generate either by the lectures or by organization, so please help to make their life easy. And welcome here at Palacký University, Olomouc, which is the oldest university in Moravia region and the second oldest in Czech Republic. And as esteemed university here, the most famous alumni is John Gregor Mendel, which you probably know. And we relatively recently had 450 years. And it was also situated here in Olomouc, which used to be a fortress city, but now it's basically a student city. And as such, it was selected as a secret jewel by the Lonely Planet. So please enjoy this part as well. Here are several important points on the map where will the where the workshop will happen. So we are here in the center of this shop at this street 17th Listopad. And then tomorrow there will be a conference dinner at the Upol restaurant, and on Wednesday we will be having the excursion to the Institute of Molecular and Translational Medicine at the Faculty of Medicine and Dentistry. And as for the program, today we have the basics. First, we will have me speaking about some short introduction. And then we will have 3 more professors. The first one will be Associate Professor Hajdúch, who will be speaking about the drug research in academia. Then we will have Johannes Kirchmair, who is here sitting in the back, who will be speaking about the cheminformatics. And then we will be finishing with the chemography concept for the chemical space with analysis by the Professor Varnek from Strasbourg, who just left out, as I see. And then we will have the poster session, and we can be at the poster session as long as you want. The posters will stay there at least till Wednesday. On Tuesday, we will be having the discussion about the structures and starting where to find the structures, and then we will move towards how to use the structures either via quantum mechanics or by the artificial intelligence by Martin Lepšík and Semyon Jesilevsky. And then in the afternoon, it will be all about the pharmacophores by Professor Thierry Langer from the University of Vienna. And there will be a tutorial. All tutorials will not be in this aula, auditorium room. It will be in a computer room in the 3rd floor. So just behind the aula. Unfortunately, we have only 35 places there, so we hope that you will fit there. On Wednesday, we will be moving towards the structure-based methods with Federica Moraca speaking about the molecular docking, and Semyon will speak about the molecular dynamics. Then we will have a guest from Bar-Ilan University, Hanoch Shenderovic, who will be speaking about the CFTR modeling. And then in the afternoon, there will be tutorials where you can learn and try these concepts hands-on. On Thursday, we will switch the gears to ligand-based methods, where Wim de Haen from the University of Chemical Technology from Prague will be speaking about the QSAR, And hopefully we will be joined by my co-organizer, Pavel Poliszczuk, who fell ill this morning. So hopefully he will be here on Wednesday to, in good shape enough to give you a lecture about multiple instance learning. If not, I suppose that he will hand the lecture to his student. It will be you, Giselle? It will be you or Alexandra? No. Not you? So Alexandra then. Who will give the lecture if Pavel will not be? Alexandra Ivanova will give the lecture then. And then there will be a lecture about the de novo design by Martin Šichov from University of Chemical Technology in Prague. And in the afternoon, there will be 3 tutorials in this time. Because there is, I forgot to mention the excursion to IMTM in Wednesday, so in Thursday there is nothing like that. And on Friday, we will have the applications, and there we will have the discussion about what innovative tools are used in a group of Alexander Domling, ERC awardee at our university at the IMTM. Then I will be speaking about the AlphaFoldology and what happens about after the AlphaFold and what are the tools you can basically use these days to where you can uptake the AlphaFold and all of the remaining stuff. Then Pavel will be speaking, hopefully Pavel, we'll see, will be speaking about his success in cage competition, where he was within the first 5 teams to find out the quality about to find out the binders to the blind testing case. And in the afternoon, we will have an online speech from the Alexander Isaev from Carnegie Mellon University. And we will be finishing with 2 lectures from the companies. First, about the drug design with NMR by Thomas Evangelidis from AIfinity, and about the cheese 3D shape comparer by Miroslav Lžičar from Deep Medchem company. So, simple, yeah? All materials I will be during the workshop, as I will get it from the lecturers, I will be uploading them to the web page. There are already materials from my talks today. And as you can see, we are streaming live, and all of that will stay afterwards at the dedicated YouTube channel and the dedicated YouTube playlist, to which you can go from the web page or via this material. And now it's time if you have any questions. No questions whatsoever? So my question, have you all registered in paper outside? Cool, because that's of course the most important part about making the workshops, to have the bureaucracy right. Any other question from the audience? Okay, if not, then enjoy the workshop. And I'll switch. And now I'll switch for the presentation of Karel Berka about drug design introduction. And the reason why we are speaking about the in silico drug design is basically that the computational methods are starting to mature to help with the drug designing process. And there is a nice quote from David C. Young from his book about that the pharmaceutical company utilizing computational drug design is like an organic chemist utilizing NMR. It won't solve all of your problems, but you are much better off with it than without it. And that's basically how the computational drug design and in silico drug designing can be seen. That is a tool that can help with the process. It won't solve everything, but definitely it will help. So, I'll be speaking shortly about where can we find the drugs. I'll be speaking about what drugs, what type of drugs were recently approved by FDA and EMA. I will be speaking a little bit about the drug design problem and what typical drug targets are targeted in drug design, and a little bit about the differences of drug design strategies. So, the first drug to be like a drug is this one, morphine. It was isolated and then used as a painkiller even for small babies to put them to sleep. As a father of two, sometimes I would be using it if I may, but somehow it's not used anymore because, of course, it had some side effects and quite bad ones as well. Since then, basically the drugs were developed more and more, and from the 50s till 70s, basically, there was the boom of drug development by the pharmaceutical companies. And nowadays, the methods changed. The precision of drug design changed as well. But, and we also changed a lot about how we test the drugs. Because at the beginning, it was tested basically on humans immediately, then on prisoners, then it was used on animals, and then we moved towards the enzymes, animal lines, and so on, in order to have the first steps of the drug design as safe as possible, so afterwards you can let the drugs only to the humans when they are properly tested. Still, the drugs are quite often based on some natural compound, but the amount of the biologics and amounts of completely synthetic drugs is slowly increasing. And these days, we basically have the drugs also as a proteins or oligonucleotides, which, for instance, we all know that mRNA vaccines were used against the COVID, and the different type of vaccination as well comes to the biologics as well. Since during this talk there will be quite a lot of vocabulary running around, so I'll go briefly through it. First, the target is the biomolecule for which you are targeting the drug. The lead is a compound which is developed, and basically the basic scaffold of the drug that is developed into a final drug. Hit basically is some kind of a compound that comes through the testing, through some screen as a positive. And from the hits, you select the candidates as a drugs that you try to develop further. And then we have two ways how to speak about the efficacy and the activity. While the efficacy is basically quantitative property that it works or not, the activity is the way how to get the number on the amount of unit which you need to have active. And the last but not least is the bioavailability, that if you have a drug which works on enzyme, well, it better work on a human as well at the end. So it has to come to the target in the target tissue, and that's the bioavailability, and quite a lot of drugs basically has to be quite a lot of co-developed at the end to basically reach the target. The drug approval timeline from the biological identification of e.g. by GWAS or something else takes ages normally. And basically in first step you try to find what your target should be, but actually recently there starts to be a little bit of shift from this perspective, because this targeted approaches quite often fails at the later stage. So nowadays it starts to be seen that maybe not just one target, but to try to find the compound first that you can develop further with the effects and to find out what the target is afterwards. But if you find the targets, if you find the active compounds, that's more important for your drug development, because then you can basically use the methods similar to ones which we will be speaking about during this workshop, like QSAR, pharmacophore, de novo design, chemical space exploration, all of those to basically find out what you can use. And then once you have this, you can try to evaluate and optimize the let's say, by molecular docking to find the spot how they fit, how the compound fits to the target, and then to find out what you can optimize on the compound further by the organic synthesis. And after that, you basically have to put it first to the preclinical testing, so first towards the enzymes and cell lines, and after that, you go through the animal studies to the participants via first phase, to second phase, first phase, and then at the end you get the registration. And this process can take quite long. The accelerated drug approval in times of needs we've seen during the COVID era. Because if there is enough of patients, you can get the statistics on the efficacy and quite more quickly than if you have just a few patients that you are trying to cure in case of rare diseases. And therefore, those phases can relatively quickly follow during the phase just by some kind of milestones that, okay, it's safe enough to put it into a second phase, and we will see the rest later on. So that was the way how the COVID vaccines were developed. Basically, within 1 year, we got the vaccines, and now they are fully registered and they are fully used. So the conventional way was quite slow, and it also had some drawbacks that the patients had to wait quite a long time to get the drug, basically. So there are, of course, several ways how you can speed up the process. The first speed up the process is that you get some kind of a rare disease, and therefore there is only a handful of people who have this disease. And therefore, to generate a large cohort of the patients to test the drug would take ages. So both EMA and FDA, Food and Drug Administration in the US, and European Medical Agency in EU, have accelerated processes for the orphans. And as you will see, it's quite often used, and it's usually used by smaller pharma companies, because you don't need large cohorts, and therefore it's cheaper to develop the final stage, and therefore even small startup companies are able to do that. Or if there is some kind of a threat, like the COVID was, of course it's possible to get a conditional market approval to speed up the process to get the drug as quickly on the market as possible. So if we compare how the amount of new entities happened during the last years, you can see that last year was actually quite good. The last 10 years, we are approximately 50 drugs per year registered by FDA, and slightly smaller number in EMA. And actually in EMA, we are on the second largest in the last 15 years almost. So last year was quite good for pharma companies' registrations. And when we look into the first-in-class, which is important because that gives us a new drug for a new patients who doesn't have anything previously to cure their diseases. These ones averages approximately at 40%. So we are still moving towards finding a new era, new patients, new diseases to be cured, which is quite good. You can see that the orphan diseases fluctuate in Europe between 30 and 50, and in US, it's around 50 all the time. So it's quite oftenly used way how to get the drug on the market. And afterwards, once you have an orphan drug, then you are trying to enlarge the registration to find out whether your drug might be used for another target or another disease similar to the previous one, and therefore to enlarge the registration further. So that's the reason why the orphan drugs are quite popular, especially in small companies. And the expedited compounds, which are the quicker ones, are of course similarly 50%, but in the COVID era, it used to be 70% were expedited. Yeah. While in the EU market, it fluctuates around 30%. So, one point with the development of new drugs is called Erum's law. It was found in 2010, 12, in this paper in Nature Reviews Drug Discovery, and it was quite worrying concept that while the computers are more and more quick and cheaper, according to the Moore's law, in pharma companies, basically you can see the downward space, where the trend was quite simply fittable by a huge decline, and this decline is number of drugs per $1 billion. So it's inflation adjusted, so it's real dollars, and the problem is that it really was going downwards, and it was really more and more expensive to get a new drug. Based on this one, based on this paper, and the analysis that the companies basically are trying to have problem with the R&D efficiency to get a new drug in the market, basically the first one was the cautious regulator problem. Another one was the management, just throw money on it and it will come up approach. And some brute forcing, not to be trying to find out what are the causes, but just the brute force the research. This problem and this trend was actually stopped at the time. And currently, we are averaging at approximately $1 billion per new compound for 10 years already. So it stopped this trend, and hopefully it will stay there. And hopefully the new methods will help here. But as the most expensive part is the testing on humans afterwards, we'll see to that. But what helped is that the innovative methods basically sped the process, and that's as such the time is money, and that helped a lot. And the production of new chemicals and new molecules starts to be easier as well, because the registrants, the FDA and EMA, basically changed a little their registration process in order to make these expedites registration, and that helped tremendously. The part is that there is actually a missing novelty in drug development. And this is quite a nice paper from the Review of Financial Studies looking into that. And here, basically, they've looked into the chemical similarity calculated by Tanimoto. Similarity you will see probably in ligand-based approaches on Thursday. And basically, they tested the new compounds based on when they were found afterwards in the same category, in this case, statins. And the first statin was lovastatin, and the similarity score to something else was just 0.25. So it's basically relatively completely random number to any other typical organic compound. Then the pravastatin, of course, since the lovastatin was there, had the similarity score just 0.6, and the simvastatin afterwards already 0.8. So you can basically look into the similarity of the compounds when they were registered to the previous compounds previously to see how often you can see a new scaffold that is used as a new drug. And if you look into this one, you can see that the similarity to previous candidates is quite often just 1. And the funny part about this one is the registrations of the compounds which had deuterated hydrogens as well, which basically you have the same scaffold, but you change the hydrogen to the deuterium, and then you basically have the same scaffold, but you have a seemingly different drug. This approach starts to be penalized later on, so hopefully this will move. But you can see that majority is that there is quite a lot long leap above the 0.6, where those compounds are actually quite similar. So the question is whether the larger companies or smaller companies came up with the new ideas. And the quite interesting finding from the paper was that the larger companies are actually more likely to engage in novel drug design, novel drug development, because if you came out with the compounds that are in this part, your likelihood of FDA approval is quite smaller than the new one, of the most similar ones. But the patent you get afterwards and the money per patent are smaller because there is already some previous drug in the area, and therefore you get lower money for the compound afterwards. While if you work in this area here, where there are novel compounds, then the approval is not likely, but if you hit the jackpot, you get the higher price for the compound. So the market is still offering the value for a novel compounds, but the companies are basically trying to go as a second similar drug to someone else had already. So when you want to design a drug, the first one to think about it in some kind of way was a lock and key analog developed by Emil Fischer, Nobel laureate in 1902, where you basically have a target, your biomolecule, and you develop a silver bullet compound that will attack to the target and you hit it. But the problem is that we already spoke that it's an expensive problem because it's $1 billion to develop one new compound into a drug. And that makes the new drugs quite expensive as well, because you have to pay for the development. And you also pay for the development of the drugs, of the compounds which did not make it into the drug. And it's also a hard problem because we have selected subset of targets. It's not really easy to find out the right pair, and you get issues with the side effects as well, with the toxicity, with the bioavailability, and so on. And as I said, the most expensive part is the clinical one. There is plenty of possible obstacles. For instance, you don't have testing model. That was hampering with the HIV, because H in HIV means human, and therefore you cannot test on mouses when the mouses cannot get HIV. So it's not really easy to test on people ethically. And if there is a rare disease, there is just a small amount of people, and therefore the question is whether they will pay for the development of the drugs. But I think that Dr. Hajdúch will speak about that a little bit more. Also, quite typical problem of the drugs these days is that, okay, you develop a drug which is non-toxic, but it's also non-potent. It does not work that much. And therefore, that's quite a hard problem. And it also depends on what type of illness it is. Most typically, when we are speaking about the lock and key analog, then we are speaking mainly about the inhibition. That we have a kinase and we want to inhibit it in order to stop the cells to divide, for instance, during the cancer. For that, you basically, from physical chemistry point of view, and I'm from the Department of Physical Chemistry, it's basically trying to maximize the binding capacity, binding affinity towards the target. But this is not the only way how the illnesses can come. Because, for instance, you can have a small response of your body to some chemical you need to have. And in that case, you basically want to have some kind of an agonist who will push your typically GPCRs to start to send a signal to the cell that something is happening. Or on the other side, if you have pain, you are taking the pain reliever, and pain reliever is usually an antagonist for these receptors. So you are blocking that ones. So again, these two parts are similar in the way that you are trying to generate the compound which will somehow bind to the target, in this case receptor. But in case of structure-based design, you have to select the structure of antagonist or agonist binder to have this part. Sometimes it can be just a regulation, and that's where the biologics come, because you can have the, for example, CGRP peptide in migraine, which basically binds towards the CFTR and helps this one, or these can be antibodies or silencing RNA. There are plenty of possibilities how you can regulate the things. And speaking about the nucleotides, the RNA interference, RNA aptamers, silencing RNA, those are also, and as you will see, relatively often used as drugs. So when we look into what are typically the drug targets, this is a figure of the drug targets by target biomolecule and their localization. Basically, 60% of those ones are somehow bound to the membrane. And it makes sense because once you have a cell, then if the compound has to just interact with the molecule, with some receptor on the membrane, then the signal is transferred by the cell itself. And the molecule doesn't have to cross the cell membrane in order to get to some active enzyme and to stop it. So in that case, you basically have relatively easier job in the development, because you just need to find the protein on top of the cell. But of course, there are more others, like the interacting with DNA itself or some transcriptor factors and enzymes, which of course are happening inside the cell. And therefore, in that case, you have to optimize the molecule in a way that the molecule can go inside. And that's actually happening with remdesivir, because remdesivir is a prodrug of a prodrug. In order to be able to cross to the membrane, through the membrane as a nonpolar compound, and afterwards it uses the enzymes within the cell in order to activate itself and transform itself into a trinucleotide, nucleotide-like compound that will then bind into the viral RNA-dependent RNA polymerase and block it. Yeah. So we have methods how to get inside the cells via some prodrugs or some drug delivery bodies like liposomes or polymers. So yeah, this can be done in order to get the drug there. But of course, it adds to the development cost, of course. If you look into the drug targets based on what type of the protein or protein class it is, the highest one is the 7TM1. These are all GPCRs, G-protein coupled receptors, and those are basically the ones who are receptors on the plasma membrane, who are taking the interaction with the drug on the top and then releasing the signal via the G protein to make a cyclic AMP and to get the information downstream. Then the second one are nuclear receptors, voltage-gated ion channels, reductases, ligand-gated ion channels, electrochemical transportase, kinases, and proteases. And you can see that there is quite a huge differences in some classes against the percentage of drugs, which is the orange one, and percentage of compounds in Campbell. And compounds in Campbell are the compounds which were basically developed as a part of the scientific papers and then deposited to Campbell with the assay data. And you can see that we have more anti-GPCR drugs than it would be against by the amount of compounds that are from the Campbell. So that's the reason that says that we have underdeveloped this part, that quite a lot, we have more drugs than the research done on that. So we will see about the CFTR modeling on Wednesday, because CFTR is one of the GPCR protein receptor that's quite important for the cystic fibrosis, for instance. On the other hand, we have large amount of compounds which were tested by academia mainly against the kinases and proteases. Basically, the kinases, that's cancer, mainly, and proteases, that's HIV. And we don't have that many compounds that made it into a drug afterwards. So this gives us the image about that some parts are actually making the drugs at the end, and some groups, it's really hard to come up with the drug after all. Yeah, and it's the amount of compounds that are newly approved, basically, in GPCR is quite quicker in registration than, for instance, in kinases. But in kinases, at the last, later on, there start to be a upheat. So I'll need to make a check-up on this one because it's from 2017. So maybe that changed a little. Then I'll be speaking a little about the differences between development of small and large compounds. if you develop small compound like aspirin here, it's basically chemistry to generate it. You can have the synthesis reproducible. You can have relatively simple identification tests and so on. The antibodies on the right side is much bigger, and it's generated by the cells. So the cells is changing their production as they go, and therefore you have to be more careful about the quality of the product afterwards. It's really harder to get the biologics as pure, and of course you need a completely different methods to get these drugs and not these drugs. For the drug on the left, you can use the QSAR because you are working with small molecules. You can look for the molecule docking because that small molecule will dock into some bigger one. You can do the pharmacophore search because you have a small molecule where you define a specific pharmacophoric groups, and you search through the database to get the knowledge about, to find out all possible compounds that will fit into the pharmacophore. While on the other side, on the right side, none of that would work because that molecule is too big. But recently, the development of monoclonal antibodies seems to be expedited by the post-AlphaFold methods, where the AlphaFold by itself is known that it generates quite nicely the single chains. And the development afterwards made the AlphaFold multimer, where you get also the complexes right, or more right than before. And this area of development develops more and more these days, and now it's possible to generate the proteins in order to get to the right shape, for instance, via protein MPNN or via chroma, or new stuff, or you can even take the drug development vice versa, and you take the small molecule and you generate the protein around it to get a sensor, for instance. So now we are more, with the AI tools after the AlphaFold, we are more successful in developing also the biologics in this part, and it will change probably quite a lot which molecules are moving towards the further steps in development. If we look into which molecules, which types are basically used are successful in the approvals, you can see in 2022 it was approximately 60% of small molecules, and from those small molecules it was mainly new organic molecules, only 2 peptides and one radio-labeled one, and that's basically quite similar to what happened in 2023. And I've checked the numbers from 2024, and it's basically the same. Still, the majority of the drugs which are approved is small molecules. And majority of this workshop is basically looking for the small molecules, so you are on the right track here. But there is growing a part which is not negligible, I would say, of biologics. Either it is vaccines, or it's proteins, or some enzyme, but majority of that is typically antibodies. Yeah. And there starts to be an amount of oligonucleotides. Well, here it was just 1, and here you can see that there are already 4. And when you check the numbers for the last year, there were again oligonucleotides approved. So this nucleotides are not coming anywhere and they are probably staying. So, my take-home message. You can generate the drug from multiple sources because the diseases are coming from multiple sources within the body. It's a hard and expensive problem, and mainly due to the biology, which is always complicated, and to the clinical trials. Most typical drug targets are GPCRs, ion channels, nuclear receptors, and kinases, for different reasons. But the orphans are moving toward the field forward to new drugs from 50%, and it's one of the most useful way if you want to generate new drug than going through the orphan way and to develop the rare diseases is a useful and vital strategy. The biologics are more complex to produce, but now, thanks to the AlphaFold, we have more and more tools to develop them and to test them further. But there is no gold path. There is not just one way how to develop the drug. Because each drug is different, and you have to look into what the problem you are trying to solve, and what compounds you have problems, and help them to overcome it. And yeah, that's it from my side. Do you have any questions? Yep.
Just curiosity question. How many sick people you need to have to be called orphan disease, and how many sick people would be needed to get adopted? If you have any like numbers.
Well, I had there a slide with the conventional way where you need thousands, which in case of COVID era was really simple to fill quickly. In case of orphan drugs, you need to have hundreds at the end. Yeah, hundreds.
But that's for testing.
Yeah. But the rare diseases are interesting area in the way that quite a lot of population actually have some kind of rare disease. And the amount of people who are sick from some rare disease is actually quite large in the population. So, yeah.
They just don't know that they have it?
In some cases, they know it a lot. In some cases, they don't. Yeah. But I think that will be up to Docent Hajdúch's lecture, where he will be speaking about that, I guess. Am I right? Okay. Any other question? If not, then thank you. And we'll switch. Oh, I'll check whether. There is no question even in chat. So cool. So now we'll switch for Docent Marian Hajdúch from IMTM of Palacký University, Institute of Molecular and Translation Medicine. And he will be speaking about the drug development in academia and lessons learned from that.
I can start before we upload my presentation, answering your question regarding the rare diseases. We have more than 7,500 rare diseases. Some of them are actually... Okay, so some of them are relatively frequent, like cystic fibrosis, but some of them are extremely rare that we have just a few cases worldwide. So therefore, even the clinical testing in those cases is limited to very low numbers of individuals. Yeah. So it's typically hundreds in things like cystic fibrosis, but it's much less in less frequent disorders.
Is there a will to develop something for those?
Yes, yes, there is. There is a lot of, typically, many of them, there are different enzymatic effects, and typically supplementation of the mutant or altered enzyme is a very successful therapeutic approach in those individuals, and it works well. And also the gene therapies are extremely successful, and many of the products are already on the market. So with the gene therapy, you basically do not wait. You don't ask for too many individuals to be enrolled. Thank you very much. But there are also disadvantages of gene therapies, and there will be always need for the small molecules, because some frequent, relatively frequent rare diseases like thalassemia, this is a very frequent disease, and it's not very likely that all thalassemia patients would have access to gene therapy, which is, by the way, already approved in upcoming years. So there are limitations. So the need there is, and there always will be a need for the small, and space and room for the small molecules. Okay, so let me switch to my topic. Thank you very much to organizers for inviting me for this speech. And I would like to discuss what are the opportunities for academic-driven drug development. And I think it's nice to see young people in the auditorium. I believe you all want to help mankind become famous and rich, and it starts in academia. Many people actually then continue to the industry, but is there any role of academic-driven drug development at all, or this is just old-fashioned concept? The outline of my presentation, I will talk about some successful case stories, about philosophy of academic drug development. I will mostly talk about unsuccessful stories and where we actually failed, and how we have learned from our failures, at least partially. I will showcase some case studies we have performed at the Institute and what we have learned. So the major determinants of therapeutic outcome and success those days in medicine could be actually summarized as a better understanding biology of the disease. This is super important because the biology of the disease brings new therapeutic targets, but also biomarkers. This is very frequently the same gene or the same protein, simply because if we are targeting the particular disease and the particular target, well, you would expect the target is present. So the target is also the biomarker, typically in the cancers, and very frequently we combined and target or design the therapy for the patients according to expression of particular targets in the tumor. We call it personalized medicine those days, and it's routine practice in cancer patients, but it's penetrating to other medical fields also. The other important area is, which is determining the outcome of the patients, is understanding the anatomical stage of the disease. This is particularly important for cancer, where you need to know whether the disease is localized or it's actually spreading over the body. And for that, we need to have a good diagnostics. But in last days, or not days, but years, it actually moved from the pure anatomical localization of the disease tissue also to testing the functional properties of the disease tissue, typically using a new molecular tracers. So you can, with the radiopharmaceutics, you can actually not only visualize the disease tissue, but you can also test whether the tissue is vital, or how it's actually metabolizing the particular particular substrate, and or whether there is a particular target in the tissue expressed. Typically, we can test the presence of HER2 receptors, estrogen receptors, and many other things using the PET technologies. So what is the role of academic drug development? Well, we are not the industry, obviously, but I think that we can contribute. We have a different role. So we can contribute to de-risking the novel drug discovery targets. Karel, a few minutes ago, actually described how risky is actually the drug development for the industry, how this is actually expensive. And that's one of the reasons why the industry is particularly targeting known drugs, and we don't have enough innovations, simply because they cannot afford failure. In academia, we are happy to fail, and at least we can learn from that. So this is a very important role of academia to develop in the development of underpinning target biology. The other area which was mentioned earlier is development of drugs for neglected and orphan diseases. Neglected are typically the infection diseases which are very frequent but not in the developed countries, and the orphan are those which are very rare and typically genetically determined disorders. So for those individuals, the pharma industry frequently doesn't have enough interest to develop the drugs and therapies because it does not make always economical sense. The other role of academia are actually the new paradigms for drug discovery, particularly to understand or study and validate undruggable targets and drug-like molecules. And of course, we are also important for access to more predictive models, but because the industry doesn't have direct access to patients, to patient tissues, we do have it in university hospitals, so we can develop different and more predictive testing models, particularly PDX, patient-derived xenografts. So if you transplant the patient tumor directly to the mice, you have a mouse avatar, and you can actually test the drugs in the mice with the patient tumor. And induce pluripotent stem cells, so basically from the blood cells of whatever Alzheimer patient, you can actually, from the blood cells, you can derive and differentiate the neurons, and you can actually study the neurons and test the drugs which can reverse Alzheimer phenotype under in vitro conditions. That's one of the applications, but you need to have access to the patients from which you can derive induced pluripotent stem cells. And we are here because we are training the young scientists here today, and that's super important. The industry is not able to train young scientists. This is the role of academia. Our institute was established years ago based on the European Cohesion Fund money. It was collaborative project with the Institute of Organic Chemistry and Biochemistry of Prague, Institute of Chemical Technology in Prague, and the University Hospital in Olomouc. And the area was, and it actually happened, to create a professional platform for development of small molecules, generically the drugs, biomarkers, and the molecular targets. So we are on the preclinical studies. We have a GLP certification for the animal trials. We are on the clinical trials in collaboration with the hospital. We have a very large compound store libraries, currently about 250,000 compounds from our collaborators, for which we are providing the testing of small molecules, but also from the commercial vendors and the open screen library. With open screen library is actually 350,000. We have one of the largest installation high throughput screening platform in academia, which is industry size and scale. medicinal chemistry department, but we collaborate with many chemists for which we are providing testing of biological activities. Under one rooftop, we run multiple omic studies and identify the new biomarkers and targets. This is because we have extensive biobanks of the disease and healthy tissues, and this results in creation of therapeutic standards. Important data collection for national and international databases. We are involved in several European open science cloud projects, including the EOSC for Cancer, EOSC Life, and some others. So creating the databases and data stores and the tools for AI in medicine. Well, so that was a short introduction, and let me start with the first case study. And this is actually the long-term project we have had that time with Professor Šárek from Faculty of Science, Charles University, later over here in Palacký University. And currently this is actually run by his colleague, Professor Urban. And we talk about the betulinin triterpenoid compounds, which have plethora of biological activities. We have studied particularly anti-cancer activity of those drugs, which are highly potent, and they have been described also for the anti-inflammatory effect. Particularly, they're activating the transcription factor NRF2 and antiviral activity, and they are actually maturation inhibitors. And we have recently published that they are also hedgehog inhibitors of the hedgehog pathway. But, so this is anti-cancer property of the compound. I'm not going into details. One of the betulinic acid derivative was actually binding to cytochrome C in the tumor cell. It was actually releasing the cytochrome C from the mitochondria of tumor cells and inducing very rapid apoptosis. However, interestingly, we have screened those molecules because we have hundreds, basically thousands of terpenoid derivatives in the library, and we screened them from different biological activities. And we have identified that these compounds, some of them, of course, there was a structure-activity relationship, very nice. They were blocking the maturation of HIV. And the maturation, they actually stopped. They were binding to the GAC protein, to the last cleavage site of the GAC protein, and it blocks the interaction of HIV proteinase with the GAC protein, and they actually stop the full maturation of the virus. The compounds with those code names were actually very highly potent in low nanomolar concentrations. They were blocking HIV maturation. They were not really cytotoxic, many of them not at all. So all these were actually very promising properties. But based on those properties, the compounds were actually licensed to small or medium-sized pharma in Montreal called Virochem. And the Virochem, during this project, we have been running a lot of assays, very enthusiastic. It was actually the fourth class of HIV inhibitors described almost 20 years ago. And Virochem was took over by the other company. In that time, they actually merged. That time, it was an unknown company. Now it's known for the cystic fibrosis products, and this new company completely lost the interest in development of small molecules for HIV, because simply that time tenofovir, the drug which was originally synthesized by Professor Holly in Prague, and some other molecules were already too good, and there was no interest in development of new drugs for HIV. And so that then actually entire process was, the drug development process was closed by Virochem, by that time the company, the new company, Vertex, which closed the program. And what we have learned from that is that the market-driven decisions are important. And of course, the merging of companies results in completely different priorities very frequently. This might be for your benefit, but also opposite, because it was for the benefit of Professor Holly. The companies merged, they returned the IP back to the institute, and they developed completely new company known as a Gilead Sciences those days. The other molecules which we have worked on were Aurora A and Aurora B kinases. These are very important mitotic kinases, which actually are important for the separation of chromatids and actually for the cell cycle. They have different roles in different phases of the cell cycle, and the pharma company we have collaborated with in Great Britain actually developed the small molecules. The company is called Cyclacells, which were in phase 1 clinical trials. And the compound was very interesting. It inhibited Aurora kinases in low nanomolar concentration. And we have been providing just collaborative research together with the Cyclacell. And we have been testing the anti-cancer activity in animal model. So the compound was actually very nicely anti-cancer. It has very strong anti-cancer activity, substantially reduces the tumor mass of the leukemia tumors transplanted subcutaneously in the mice, but also the infiltration of the bone marrow. There was a very nice anti-tumor effect. Also, consistently with the mechanism of action, the compound induced accumulation of cells in G2M phase of the cell cycle, particularly in G2, where the Aurora kinases are typically important. And also, there was increase of accumulation of polyplate cells, so more than G2. And those cells were actually accumulated in dose-dependent manner, and this was also consistent with the mechanism of action, because Aurora kinases are important for this process. During the histology examination of treatment tumors, I have seen interesting phenotype that there was a lot of the density of the microvessels in the tumor was substantially reduced in the treated groups. When we stained the microvessels with anti-CD31 immunohistochemistry staining, you see these brown microvessels, very dense in the controls, but very rare in the treated tumors. And this was interesting that time because it was not well understood what is behind it, but we suspected the molecules and targets involved in angiogenesis. And we actually sent the compound for the screen, for the kinase screen, and it came out that this compound is not only Aurora kinase inhibitor, but it's also inhibitor of vascular endothelial growth factor receptor, which is very important kinase, which regulates angiogenesis in tumors. And then, of course, the company came with the new target, so it was very nice that you have a compound which blocks both kinase activity, but also the angiogenesis, and they were actually able to sell it with better preclinical information. So that was an interesting story, and this is also important from the point of view that for the good anti-cancer activity, you usually need to hit more than one target. Very typically, super-selective kinase inhibitors or super-selective inhibitors of one target are usually not performing well in preclinical and clinical trials. Okay, so what we have learned, that the collaborative research with academia, actually the company learned, has the added value, because in a commercial CRO, they would just test what they asked for, but we are coming with more kind of scientific approach, and usually we are trying to understand mechanisms which are behind the actions of individual molecules. The other project, the other case I would like to show you today is the development of paclitaxel prodrugs. Paclitaxels are actually known mitotic inhibitors. They block the polymerization of tubulin, so they basically stop the cell cycle of the cell on G2M transition, and the cancer cells stop cycling, and they eventually die by apoptosis or some other mechanisms. The paclitaxel is a highly hydrophobic molecule, as you know, and these hydrophobic properties are actually limiting the clinical use of the drug, particularly because you have to formulate the drug in a very aggressive formulation, which is composed of chemophore. Chemophore is kind of Tenzit, which is like any other Tenzit you use in the dishwashers. It's not doing very well to the patients, so you have to pre-medicate patients with corticosteroids. You have to give them a lot of antihistaminics, so they do not have reaction to chemophore. And the idea was to develop the more hydrophilic prodrugs. We have synthesized in collaboration with Californian company Biophysica Foundation the paclitaxel prodrug, which was actually polyol carbonate, which in the water solution decomposed to paclitaxel and the small molecule. So there was very nice kinetics. We have tested preclinically. The code name of the protaxel was BP179, and it was substantially more potent in terms of efficacy than paclitaxel. In red, you have a paclitaxel. In yellow, you have a protaxel. It was substantially more potent. It was even active in paclitaxel-resistant tumors, interestingly. For instance, this is myeloid leukemia cell line K562, xenotransplanted to the mice. It's growing very well. If you treat it with a paclitaxel or protaxel, there is complete elimination of the tumor. But if you have a paclitaxel-resistant cell line, K562 taxol-resistant, which is overexpressing pharmacological transporters like MDR protein and some other, which can actually pump out the anti-cancer drugs from the cells and clean them, the paclitaxel is not effective compared to vehicle, but protaxel was still effective. So this was an expected observation that we try to understood, and we have understood and shown that actually the paclitaxel as a prodrug is blocking the pharmacological transporters, particularly MDR1 protein, and so the prodrug was actually chemosensitizing the tumor cells, and the released paclitaxel was entering the chemosensitized cells where the transport and efflux of PGP or MDR1 gene was actually blocked. So that was the mechanism of action. And the compound was developed up to phase 1 clinical trial, which was performed here. It was a single center open study, and we selected or pre-selected for phase 1 trial the paclitaxel refractory or relapsed ovarian cancers or prostate cancers. The idea was, of course, in the phase 1, you always, this is about the safety, it's not about the efficacy, but efficacy is nice secondary endpoint, particularly if you want to sell your product to the industry. And the other area was anti-androgen resistant prostate cancer. We performed intrapatient escalation, and the primary endpoints, as I mentioned earlier, was toxicity, tolerability, maximum tolerated dose, and dose-limiting toxicity. And the secondary endpoint was efficacy. The compound was very well tolerated and safe. I'm not going into details of that, but it was also very efficacious. You see, like this is one of the ovarian cancer patients, and over here the patient started with very large pleural effusion, which during therapy almost disappeared, with the tumor markers disappearing after 5 cycles, so the compound was very nicely active. Also in the prostate cancer individual, this is before chemotherapy, multiple, this is bone scan, these dark dots, this is the urinary bladder, but the rest, the other black spots are skeletal metastases, and they were substantially reduced in number and also in the size after 5 cycles of chemotherapy, with substantial reduction of PSA as a tumor, as a prostate cancer biomarker. So this was extremely successful. We started the negotiations with the companies to license this molecule, but we actually faced one of the major problems we have in the drug development, and it's that we need to publish or perish. And in this case, it was publish and perish because the patent was filed too early. So typically, just the target discovery takes around 2 to 3 years, lead discovery 2 years, advanced preclinical analysis altogether 6 to 7 years, and the clinical development is minimally 7 to 8 years. So at the end, we came to the industry with a very nice product with proof in the clinical trials, but there was no interest for the licensing, primarily because there was not enough time to develop the product on the market with patent protection. So what we have learned is that time is money. And from this time, we are trying to patent the molecules as late as possible. And the most promising molecules we patent or file the patent when we actually enter phase 1 trial. Unless you are in the super high risk that someone else is working exactly on the same target or the molecules, and you are in the risk that you would actually lose your IP. Yeah. The other case report we actually worked on was developing, that was actually done with the same company. We have been trying to develop the effective therapies for prostate cancer. And you know that prostate cancer is androgen dependent. And those androgens and the blockation of androgen receptor is super active therapy in the prostate cancer. One of the most potent androgen derivatives, which is actually on the market and in the clinics, is flutamide. And we have been synthesizing the derivatives of the flutamide. Flutamide is a competitive inhibitor of androgen receptor, and we wanted to synthesize the covalent inhibitor of androgen receptor, which we hoped will be substantially more potent. Well, we were not successful, but we have synthesized this strange molecule we called later fluoridyl, which was super potent androgen receptor antagonist, but it was extremely unstable in water. And we have realized that this would be a fantastic opportunity to develop it further for androgenetic alopecia, because this process is also androgen receptor dependent. Typically, the expression of certain types of androgen receptors in the hair follicles is actually preventing growth and regrowth of the hair, particularly in men, but also in females. And the interesting opportunity for this molecule was that it's extremely hydrophobic. It's very stable in the lipid, but very unstable in the water. So the idea was that this hydrolytic instability and quick disintegration to the fragments with no hormonal activity would actually provide a competitive benefit to this molecule during the topical administration, because from the topical administration to the hair scalp, the drug, even if it's resorbed, it would be actually immediately decomposed to hormonally inactive molecules, so it would not decrease the libido of the man. So that was very important that time, because there were a number of molecules, anti-hormonal, which were effective in growth, in the hair growth, but they were actually also decreasing the libido. So this molecule was brought to the clinical trials, and the clinical trials we have been testing the number of anagens and telogens. Anagens are actively growing hairs. Telogens are kind of hair which is being replaced by the new anagen hair very soon. And the compound was quite active that time in the clinical trial. It was very nicely decreasing the number of telogens, increasing the number of anagens. It was placebo-controlled trial. And based on that, the compound was actually brought on the market, and it's still on the market under the name Ukapil, and you still you can buy it. So this is on the market for 20 years, almost. No, it's actually probably less, but it's still the only one topically active anti-androgenic molecule which is used in cosmetics. So what we have learned from that unexpected drug characteristics may result in new product and or indication. And we also gain some valuable expertise in development of cosmetic products. So sometimes you work on the drug for the prostate cancer, and it came out the cosmetics for the hair growth. But take your advantage. The other example is from drug repurposing program. At the Institute, we have a very large drug repurposing program. I will showcase today only 2 examples. One is fenretinide, the other one will be about disulfiram. Fenretinide is a synthetic retinoid. The compound was used or developed for the treatment of pediatric neuroblastomas, with the idea that it will actually increase the differentiation rate of the tumor cells, and it will actually result in regression of the tumors. But the compound was also extensively used for the chemoprevention trials. So thousands and thousands of individuals were using the compound for years. So we have a very nice safety data that is very safe. But at the end, it was not very efficacious in terms of prevention of cancer, particularly breast cancer. There was some borderline activity in the young breast cancer individuals, but not in other indications. There were also sporadic reports on anti-inflammatory activity of the compound in rheumatoid arthritis, psoriasis, and we have published actually the first papers that the compound is very active in cystic fibrosis. The cystic fibrosis is a rare disease, that's what we have discussed earlier. And you will come back to the CFTR gene and with your chemoinformatic work, so it will be probably the interesting introduction. That time, it has been known that the children with cystic fibrosis, the major problem they have is the colonization with the bacteria. Typical problem is Pseudomonas. They are colonized, they get reinfected, and they eventually die because of the progression of infection diseases. So they are not primarily dying because of the rare disease, but they are dying in young age because they are not able to resist and clear the pathogens from the lungs. One of the reasons why they cannot actually clear the pathogens is different composition of the lipid monolayers or bilayers on the surface of respiratory cells, but also in other cellular membranes. It has been shown that the CF patients, but also CF heterozygotes, which do not have a clinical symptoms, typically parents, they have a lipid abnormalities, particularly they have very high concentrations of arachidonic acid in the lipid membranes and very low concentrations of arachidonic acid is pro-inflammatory fatty acid and very low concentration of decahexanoic acid, DHA, which is anti-inflammatory fatty acid. And this actually results in a very massive and easy and a very low threshold for the induction of inflammation in those individuals. And this pro-inflammatory status in the cystic fibrosis is inducing the inflammation and fibrosis, because fibrosis is consequence of inflammation. And this is what is actually killing the patients. It's lung fibrosis, but this is because of inflammation, and inflammation is there because there is infection. Yeah. And so we try to identify the compounds which could actually normalize this status, shut down the inflammation, and basically to improve the lipid profiles in CF patients. we have developed the models of CF mice, which recapitulates the disease phenotype. And those mice, they did have indeed very high CF mice, very high concentrations of arachidonic acid. This is wild type with normal genotype. And if you treat these CF mice with fenretinide, if you treat wild type mice, there is no difference. But if you treat the CF mice with fenretinide, there is a rapid decrease of arachidonic acid. That actually was very consistent with inhibition of inflammation. On the opposite, there is also, this is the DHA, anti-inflammatory fatty acid. Wild types, high concentration, CF, almost no anti-inflammatory fatty acids. If you treat those CF mice with fenretinide, you increase the anti-inflammatory fatty acid content. So this was interesting observation, which triggered a lot of studies, and including mechanistic studies, and we understood that there is also increase of ceramide or decrease of ceramide metabolism. We have identified several molecular targets, fenretinide, which was actually later licensed to the Lauren Pharma and renamed to Lau7B. I will come back to that later. And the compound we shown and published several papers is actually inhibiting desaturases of fatty acids and ceramide synthesis. What is important that when we wanted to launch the phase 1 clinical trial in Canada, it came out the problem with prion infections, with the mad cow disease. And the problem was that the beef gelatin was prohibited as a component of the capsules. So until that time, vast majority of capsules, gelatin capsules, were actually done or produced from the bovine gelatin. That time it was not possible. And all capsules which were available at NIH, well, it was possible in the US. In the US, they were still allowed to use the bovine gelatin for the formulation of drugs, but that was not eligible anymore in Europe and in Canada. And it was, however, lucky coincidence because all the drug stores, fenretinide stores, which were suspended in corn oil, in the bovine gelatin capsules, were actually at NIH repositories, and we could not use them for the clinical trials in Canada and in Europe. So that was like the end game. So then we decided to reformulate fenretinide instead of suspension in corn oil, where the biological availability was very low. We developed completely and patented completely new formulation of fenretinide, which was reformulated. The drug was actually manufactured in porcine gelatin capsules, so it was perfectly compatible with the clinical trials, and we could actually launch the phase 1 clinical trials. That was an interesting experience because without having capsules in bovine gelatin, we would not reformulate the drug, and we would never gain any intellectual property and opportunity to develop it as a drug for the patients. So phase 1 trial with reformulated compound went very well. It was safe. It actually improved the lipid profiles as we have described in mice. So this was actually confirmed, or the lipid pathway was confirmed. We also demonstrated the decrease of ceramide 5 synthesis, which was consistent with previous studies. And the patients which were actually on medication with Lau7B, they increased the forced expiratory volume, so the decline of their lung functions was much smaller than in patients who were actually not having fenretinide in the medication. So it was very nice outcome. The drug is currently after phase 2 clinical trials, and the company is trying to launch the funds for phase 3. During the COVID pandemic, because the compound has very strong anti-inflammatory effects comparable to corticosteroids, but it's not immunosuppressive, it was actually tested in COVID individuals, and we received also fast-track approval from FDA, actually for the individuals with the with the COVID with medium severity. And the study was closed, and I think it's already published. And the study demonstrated that patients medicating fenretinide after admission to the hospital, none of them was actually mechanically ventilated. So it was a pretty nice outcome. Okay, so what we have learned? There are differences in US, EMA, and Health Canada regulations. So there are small regulations, like one regulatory authority approves bovine gelatin, the other one does not. But these small differences may have substantial impact on your drug development. So the new reformulation strategy substantially improved pharmacology of the compound. We improved the resorption of fenretinide about 10 times, and this actually resulted in new intellectual property and the spin-off and the development of the molecule towards the clinical applications. And fenretinide is an example of triple drug repurposing, particularly in COVID, because it was developed for treatment of cancer, from cancer to CF, and from CF to COVID. So you can repurpose the molecules up to infinity. I think that the world champion would be metformin those days, which is repurposed for so many indications. Okay, the other example from our drug repurposing projects is disulfiram. Disulfiram is a well-known molecule for the treatment of alcohol abuse. The compound was developed in Denmark, and in the 50s, and the molecular mechanism is actually the inhibition of acetaldehyde dehydrogenase, and this inhibition results in accumulation of acetaldehyde after drinking alcohol, which is a highly toxic molecule and which actually induced a lot of adverse events. So simply, alcoholics who are medicated with disulfiram, they cannot drink. They have a very serious health problems. Early after introduction of this compound on the market, there were actually reports, anecdotal reports of anti-cancer activity, including the complete responses of breast cancer patients reported in 1956, and metastatic case with a very long duration. They described it as a spontaneous regression of breast cancer. And this has been reported several times, and such reports were very frequently reported, and the patient was on therapy for 10 years, and she was doing very well. So we wanted to confirm those, systematically confirm those anecdotal reports, and we performed a large-scale epidemiological study using the drug and epidemiological registry, this time again in Denmark, in Copenhagen. And from the Denmark cancer registry, we were able to download the information from patients suffering from cancers, from localized cancers, and also disseminated, non-localized stage of the disease, from those who are actually using, together with other anti-cancer medication and treatment, as a continual users of antabus, and those who actually stopped antabus with anti-cancer therapy. And with those epidemiological studies, these were actually hundreds, thousands of records, it came out that the continual users who actually continued disulfiram medication, they had very dramatic reduction of the risk that they will die. So it was reduced about 40% overall, 69% for the localized stage, 71% for non-localized stage. So this was really a very impressive reduction. Rarely you have anti-cancer drugs which could actually decrease the risk of death or disease progression 30 to 40%. That's very, very rare. So we started to study what is the mechanism of action. It came out that it's not directly disulfiram. Disulfiram is actually a very unstable molecule. It's decomposed to two molecules of diethyl-dithiocarbamate. Diethyl-dithiocarbamate is a very strong chelator. It chelates the copper in the body immediately, preferentially copper, with very high affinity to diethyl-dithiocarbamate copper complex. And this is actually the molecule which is responsible for anti-cancer effects of disulfiram. So one of these, this is the anti-cancer metabolite, one of approximately 30 metabolites of disulfiram, which is responsible for anti-cancer activity. We also identified the molecular target, which is NPL4 protein. You can read it in the publication. It's actually the protein important for translocation of misfolded or mutant proteins from endoplasmatic reticulum to the cytoplasma, where they are degraded by proteasome. And this is very important pathway for the cancer cells. If you stop it, the cancer cells start dying. There is there is also the window of opportunity, because in the normal cells you have much less damaged. You don't have mutant cells in the normal cells, so there is much less misfolded proteins, and those misfolded proteins are needed to be degraded. Otherwise, the ER is actually stuck with misfolded and damaged protein. And this is basically the induction. This results in induction of ER stress and proteotoxic stress. Interestingly, however, still the disulfiram is inhibitor of acetyl dehydrogenase. So for the cancer patients, it was not really appreciated side effect that they could not have a glass of wine in the evening. So this was very frequent reason why they actually stopped using disulfiram for anti-cancer indication. However, we have published that this diethyl-dithiocarbamate copper complex is not inhibiting aldehyde dehydrogenase. It was published in Oncology in 2019. So it has all good properties. So it's a direct anti-cancer molecule. It's not inhibiting the acetaldehyde dehydrogenase. So we wanted to develop it as a drug. But the major problem was very low solubility. The solubility of diethyl-dithiocarbamate in the water is about 0.5 nanogram per milliliter. But if you formulate it in albumin, it dramatically increases to more than 10 milligrams per milliliter. That's a substantial increase of solubility. And what was behind that? Behind it was a lot of research, but at the end we understood that this is actually the formation of nanoparticles of diethyl-dithiocarbamate with albumin. They have been characterized, so these are composite nanoparticles which can be manufactured in very fast, very easy way with very high reproducibility. It was part of the patent which is awarded now, and diethyl-dithiocarbamate albumin nanoparticles are actually entering the phase 1 clinical trials, most probably this year or early next year. The clinical batch has been already manufactured. So what we have learned? We have learned that good science is important. We have learned some skills for nanomedicine product development, and also that regulatory advice may guide further development pathway, because the regulatory advice was very important to talk to the regulator how the drug, like a nanoparticle formulated drug, will be developed, because it's important to find a mutual agreement with the regulator. And also the contraindications may give you a competitive advantage, particularly because antabuse is not compatible with alcohol. Many of the drugs are actually dissolved in alcohol, so you cannot use the medications which are formulated in alcohol in the sulfiram users, but you can do that with COET users. Okay, the last class of molecules I will talk about today are tracers and radiopharmaceutics. As we discussed earlier, the anatomic localization or the anatomic understanding of disease stage and localization is very important in clinical medicine. But those days we can not only identify the localization of the disease, but also to test the functional properties of the disease tissue. I will skip from cancer to infections again. And these are very close entities because infections are particularly frequent in cancer patients, and many infections are actually causing cancer. The infections are the most frequent cause of human cancers, typically HPV, gastric cancers, and some others. So the identification of tracers which would visualize the site of infection are very important. Why? Because in many clinical situations, particularly in sepsis patients, in newborns, in many situations, we don't know where is the location of the infection, where it is located. And it has important clinical impacts. So early and accurate diagnostics is often essential for successful treatment. And the current methods which are used are typically based on the PCR or cultivation of pathogens are not good enough, simply because many of the pathogens, like at least 40–50%, you cannot cultivate. They will not grow up under in vitro conditions. The PCR is a very targeted method, so you have to know what to test. And you can miss, if you not target properly, you can actually miss the pathogen. So we need something what is more non-specific, but still specific for bacteria or viruses or for particular pathogens, but reasonably broad that you can actually detect a broader spectrum of infections. And these are actually siderophores. Siderophores are low molecular mass iron chelating molecules, which are typically used as iron transporters. The iron is absolutely essential for all microbes. They cannot live without irons. If you have them in iron-free conditions, they die very fast. And they even developed bacteria, fungi, and some other microbes. They develop particular iron transporters, siderophores, which are released to the environment. They chelate iron in the environment, and they are proactively transported back to bacteria to grow up, because the iron is needed for the growth and proliferation. So, so that's one part. The other part is, of course, the iron. The iron analog, iron doesn't have isotopes which we could actually use for imaging and tracing. But very close to iron is gallium, and gallium has fantastic isotopes which can be used for the PET imaging. So typically gallium-68 is the suitable radionucleotide for labeling the siderophores. And you can basically, with gallium-68, beautifully cheat bacteria. You can load the siderophores with gallium-68 instead of iron. You can expose them, bacteria, to this Trojan horse complex, and they will actually eat it. And once they will pick it up from the media or from the tissue, they will start they will flash on the PET scanner. Sorry. So basically, what we used germanium gallium generator, which is clinically used source of gallium-68. And we have combined it with a compound which is used for chelation of iron during the iron overload diseases, like some anemias, for instance, which is called desferal. And desferal is originally desferoxime B, which is indeed bacterial siderophore. So if you, this drug, which has been used for the treatment of iron overload, particularly anemias, combined with existing gallium generator, you have two FDA-approved compounds, and you can actually test this gallium desferoxamine B complex for imaging of bacterial infections. We run several animal models. Many of them have been published across different disease or infections. This is mouse PET-CT, where you see very nice accumulation of the desferoxamine B gallium complex in the site of infection, in the muscles, also in the lungs. This is the excretion of the tracer through kidneys to the urinary bladder. So that's okay. So conceptually, this worked very well. And the compound was accumulated in the sites of infection, and it actually very nicely enabled location of the infection. Not only location, but if you would treat this pneumonia with antibiotics, the uptake of siderophores would actually become completely inactive within 24 hours if the antibiotic works. So it's also good for testing of response of individuals to particular antibacterial therapy. Once we had all the data, we entered the clinical trial. The clinical trial is ongoing at the moment in Innsbruck University, simply because in Austria the regulation for the clinical trials on the tracers is much simpler and straightforward. There is not required the full GMP in Austria, unlike in the Czech Republic, but that's advantage of the Germany and Austria they have for the development of tracers. And indeed, the first patients, already the first patient with osteomyelitis demonstrated very nice accumulation of gallium desferoxamine B in the site of infection. This is fluorodeoxyglucose accumulation, which is a classical tracer, which is, however, also uptaken by tumor tissue. It's primarily used for the cancer diagnostics. And you would notice also very high uptake of the glucose in the brain. Well, the brain needs glucose, but not the desferoxamine B. So over here you can use this particular tracer also for the imaging of brain infections. So what we have learned? We have learned that an expected difference in regulation of radiopharmaceutics in EU versus Czech Republic, particularly in Austria versus Czech Republic. And we gained valuable expertise in development of radiopharmaceutics. GMP certified manufacturing site over here in Olomouc is currently under preparation. So we have constructed the GMP manufacturing site, and it will be certified this year. It's already in the process. Excuse me. Okay. At the end of my talk, I would like also highlight two important Czech contributions to nuclear medicine and radiopharmaceutics. The fluorodeoxyglucose, the most frequently used tracer worldwide, was synthesized and published in 1969. And in 1990, It was approved by FDA for the PET. It was, or the original synthesis was described by Professor Pacak and Czerny. And Martina Benešová recently succeeded with the development of lutetium PSMA complex, currently on the market, FDA approved, and on the market name Pluvicto since 2022. And this is used massively for treatment of metastatic prostate cancer patients. This is a very successful story. Okay. So at the end, academic drug development, yes or no? I hope I have convinced you, yes, but what is important? As always, good science is important to raise interest. You need to have a clear and logical plan to development of a product, strong team of dedicated and experienced scientists, positive attitude, and do not get disappointed because the frequencies of rejections and failures during drug development and licensing process are much more frequent than in the publications and grant approvals. So I thank you very much for your attention. And the last word of Winston Churchill, success is going from failure to failure without loss of enthusiasm. So I wish you a lot of enthusiasm for drug development. Any questions? We have a few minutes to go, but I think you can use it more wisely. So thank you very much, and see you one day in the drug development process and pipelines. Thank you.
Welcome everyone to the third talk. I'm really grateful to the two previous speakers who gave a very nice introduction to drug discovery already. I do have a few slides on those too, but I will keep that more on the surface then. Before I start with the actual topic, I would just like to very briefly tell you a little bit about the background of our group, what we are doing, and this word cloud should already give you some ideas of what we have been busy working on. So this is some keywords generated from our group's publications. And we are based in Vienna since 2020, a growing group. I personally have a very strong background in computer-aided drug design, and we have been moving more and more towards machine learning. And I'm very happy about this team here. It's extremely diverse, not only in terms of their geographical backgrounds, but also in terms of their scientific background. So we basically cover anything from math via chemistry to informatics. And these people are actually quite happy, in particular if I'm not in the frame. You see, we are having a lot of fun. So what are we doing in Vienna? We employ and develop computation methods that enable us to answer questions in the context of drug discovery. So we don't have like our workhorse method, or we always want to converge on machine learning. This is not what we are doing. We basically are really open-minded in terms of what we are doing and what software and what algorithms we use for that. So very often, people just knock at our door, scientists, and they have some questions like, I'm a crazy chemist, I do crazy chemistry, but I have no idea of what to do with my molecules. Or others having a very specific assay, they have a need, they have identified a target that is related to a disease, they want to target it, and they want to come up with molecules. And we are connecting these two words and other words together with computational approaches. So we do a lot of screening. By now, you can purchase fairly easily compounds on demand in the range of several billion compounds, which are actually purchasable at a rate where universities can afford a few compounds. And hopefully, if your predictions are right, you don't need more than a few compounds. So when we have this typical like small-scale academic drug discovery project, like a bit of the size of a master study, there should be no problem actually to purchase 20 or 40 compounds and then to test them. But the real work is not just finding a bioactive compound. Finding the bioactive compound can be a piece of cake compared to what else you need to consider in drug discovery. So it's more about just the activity. You need to think about selectivity, toxicity, bioavailability, all these pharmacokinetic aspects. And there we are very active, and we're actually doing quite a few approaches in the context of machine learning there. And what I would also like to emphasize is that there's very few projects in our group where we don't have the direct contact with experimentalist groups. So we are very, very much involved in working with companies and also with other academic labs around the globe to basically get really a confirmation that these models work prospectively. And we are about to launch a new web server. This is now the development alpha version, so this is not basically the final version yet, but the final version will go online within the very next few days. And I think this could be very interesting for you because you can consider it something like the Netflix for cheminformatics. So this is a very scalable, easily maintainable platform where you can just plug in all your machine learning models and you can run them in-house on an infrastructure which you can basically expand, and it's expandable and flexible like what you know from like the most relevant cloud services out there. So this will be launching soon, and we will also have a publication and the source code and everything available, so that could be really nice for your groups to also make models accessible to the scientific community. So now coming back, this slide in a very similar tone you've already seen. Basically just saying drug discovery remains a risky and time-intense process. And I actually prefer this slide. I saw this once in a very nice presentation from researchers from OpenEye, which shows a bit just how chaotic and multi-step and complex this journey is to a drug. I want to emphasize one thing. You can never trust in having one compound. So academics, typically academic groups or like smaller research groups that jump to the roof in front of joy that they have found a small molecule. The chances that this ever turns into a drug or is developed in a drug is still very low. So you need to always bet on many different horses. So one of the key points is really we need to make more compounds. And what we can also not trust is in a single assay. We need many assays. We cannot even trust a single animal model often to basically really understand whether a compound will ever go in the clinics. And getting here is still comparably easy. So this would be basically the point where you have a bioactive compound. But then is where still lots of problems can occur. Of course, anything that is related to bioavailability, to toxicity, and many other pharmacokinetic aspects. Medicinal chemists are the experts. They are really good in overcoming these challenges. So all the path towards the drug is really facing one challenge after the other. And that usually, or can often pan out well, as long as the principle that is responsible for the bioactivity, so the pharmacophore, is not overlapping with the toxicophore, because then you have a problem, right? So basically, if your compound has the pharmacological activity for the same reasons than it has the toxicological activity, then it's very difficult to get out of this. And if you can fix this, eventually you get to the clinical trials, which on the upper end can cost many million euros, but hopefully you will then end up with your drug molecule. In total, and these are some old numbers, but newer ones seem not to be out there really, so you need on average about 400 scientists that work for 15 years to develop one drug. And by the way, this is only the average number of costs for a new drug where the failures are not calculated in. So to look at this, nowadays, drug discovery, to most people, flavor is still too chaotic. It depends very much on the entry point where you end up with your molecule. You can have two teams of scientists, even in the same company, and there's a high likelihood they will end up with completely different active compounds. You can have two computational chemists using the same method. They will also likely come up with completely different compounds. What we actually want to have is an efficient way to come up with drugs. And this is basically one of the main ideas or aims where we are aiming at, right? We want basically computational approaches that combine all the expert knowledge and get us straight forward to the optimum. Whereas in drug discovery, we don't even know if that point exists, right? We are not sure. We cannot really be sure about that, which is very different from designing the Cybertruck, right? If you design the Cybertruck, you know, if you put in 1 billion, then it's likely the Cybertruck comes out. If you put in 1 billion into drug discovery, you still need to be very lucky that the outcome is what you wish for. So all these perspective that we're now trying to build is to have something like an automated or guided approach that brings us directly to the sweet spot, to identify the compound that needs to tick so many different boxes. So again, it's not about bioactivity. This is one challenge out of many. And this is where computers come in. In drug discovery, basically every single step in drug discovery nowadays can be supported and is in reality also supported by informatics approaches. Sometimes people have a bit of a hard time to distinguish between these individual fields. It's actually fairly straightforward, but there are some gray zones in between, right? So the bioinformatics is something where you would say this mostly concerns with genomes, with genes, with proteins, protein structures on a large scale in particular, and a subsection of it is structure-based bioinformatics, which then overlaps on the other hand again with cheminformatics. Cheminformatics is anything that has to do with chemical information, chemical data that you need to store, to organize, to manage, to retrieve, to analyze, to visualize, as we will see with Professor Varnek's talk, who is on next. And today, the main driver are different angles of machine learning. Besides that, we have molecular modeling, which is really the modeling about structures, small molecules interacting with the biomacromolecules. And this term usually relates to faster, less computationally expensive methods, so force field-based methods. Whereas computational chemistry mostly refers to modeling physical-chemical properties with computationally more expensive quantum chemical approaches. But these areas, there is some gray zone in between. Machine learning makes quantum chemistry faster, and so on. So there's a lot of development going on. There's also several definitions of chem or chemoinformatics out there, but as I already mentioned, it basically encompasses everything that is related to making chemical information usable. By the way, the slides are very verbose. They are not meant to support the talk. They are primarily meant for you to have later on a reference if you're interested in this. So don't try to read everything. It's really meant for you for later on to have a reference. So why should we do chemoinformatics? Obviously, as we know already from the previous two speakers, there is an urgent need for more efficiency in drug discovery. So the indicators are pointing up. We are hopeful, and we have all reason to be hopeful and encouraged, but more efficiency is always nice. And just to mention, the chemical space is really of enormous dimension. Think of what we know about drug-like chemical space. That could be something that's, like a salt grain compared to planet Earth. I always like to show these numbers. They are from, actually from Pavel Polishuk. He, together with Alexander Varnek, Professor Varnek, they extrapolated the approximate size of the chemical space. And when I already had the observation that when I asked students, so what do you expect? How large is the chemical space? At some point, the student said, 10 to the power of 30. I said, maybe it's 10 to the power of 60. And students were very encouraged and say, oh, that's just about twice the size as we estimated. It's of course not. So these are unimaginable numbers. These are numbers where we're talking about higher numbers than we have particles in the observable universe. So there's no way we can systematically explore those, right? So we need some heuristics, some really smart approaches to make use out of these. So what else? So in the public domain, there are about 5 billion compounds that robots are very likely to synthesize immediately. So for example, enamine or Wuxi, they have libraries where you have like a 70 or 80% chance that these compounds can be very quickly synthesized and delivered. On stock depends on how you define it and how big your purse is, but you can order easily about 8 to 12 million compounds. They are like just off the shelf available, and you have a bit more money, and you can wait a bit of time, then you can get like tailored compounds in the range of about 230 million compounds. Known natural products, some authors claim it's in the range of 700,000. In reality, it will be a bit lower because the data sets are admittedly quite still messy and contain also compounds that look like natural products but are actually natural product derivatives. So this is about the range of numbers and molecules that we are dealing with. And computational approaches, they have been incorporated in all the phases of drug discovery. Think of computational approaches not necessarily as a problem solver, but what they do is they scale down the problem to a range where you can actually address it, right? So basically we have this incredibly huge amount of data, and what computational approaches can do really very well is to point you towards the most promising compounds, like something like really like a cherry-picking approach, if you wish. And we've already learned in the previous talk about this importance in academia to really identify a new target. There, computational approaches can help a lot to identify viable drug targets, assess or identify binding pockets that are druggable. Then later on in hit identification, there are really powerful tools out there for virtual screening, for de novo design, generative chemistry. This is like very, very, very hot areas of research. Then once you have the hit, once you have a compound that triggers a positive signal in your assay, a hit is not necessarily more than that, this is where actually the big work starts, right? So basically you have this multi-objective optimization problem where you eventually want to optimize really many different properties to come up with a drug that has acceptable pharmacokinetics, acceptable pharmacological activity, and an acceptable toxicity profile. And computational approaches can help greatly in that because as you go further, you collect data. And as you collect data, data-driven methods, such as machine learning, can benefit greatly. And the further you go here, the game changes. The game changes because you get more data, so more different approaches become applicable. So this slide contains one or two key messages. The first key message is the method that you can use always depends very much on the data that you have out there. It matters a lot whether you have basically valid starting points, such as known substrates that you want to basically turn into an inhibitor, or you have a good idea of the protein. If you have nothing, then it's a bit tricky, even for computational approaches. Then what you can do is, of course, to look at related proteins, try to learn from that, or filter your database for physical chemical properties, or at least work with some diverse set of compounds. But however, more and more AlphaFold and so on, or models that basically aim to understand the whole knowledge of chemical biology, more and more we will have hints about our protein, our biomacromolecule of interest. And this is what we consider as structure-based approaches. So structure-based approaches in cheminformatics, these are approaches that make use of information on the biomacromolecule. So structural information on the biomacromolecule. They can, of course, also include ligand information when available. And the ligand-based approaches, they focus really on the chemical information. For example, known ligands, known substrates, known inhibitors, and so on. And of course, the best is if you have information on both domains. So now, by just thinking about, let me ask you that question. Who would prefer to work with only molecular structures of the small molecules? And who would like to start, or who would prefer to start their drug discovery project from a protein structure? So who is for the small molecules? 4, 5, 6, something like this. And who is for the structures? A little bit more. And a few, or the majority is undecided, as it often happens. Yeah, so the best is, of course, you have both types of information. But the reality is not that one has a clear advantage over the other one, right? So it's basically, if you have the small molecules, you have one big benefit. You know they work. So you know basically they are able to approach the biomacromolecule and inhibit it or modulate it. Whereas if you have the structure, you have much more knowledge about your target, like what it looks like. You may have a very much more direct angle towards what it looks like and which route you need to go for optimization. But because the structures are more complex, you need to also use more abstract representations in some occasions to represent the molecule and perform some screening. So at the end, it's not so clear who will win the game, right? And so what is basically the consensus or the consistent observation across the field is there's no such thing as the best method or the best approach. You see this always and always again, that the winner is always a whole ensemble, a variety of methods that come together. And this is why, this is because all these individual methods, they are able, there's like cherry pickers, right? So they have their own domain, their applicability domain, where they work particularly well. And there they can cherry pick and identify a subspace of interest. So if you have like two distinct methods, each of them may be able to identify 10% of what's out there to catch, okay? But if you overlap the hits, the overlap will be surprisingly small. And this is the thing. So basically this means rather than breaking your head about which method should I use, which one is always the best, this is not the right question to ask most of the time. Most of the time, the right thing to do is just use anything. Just use things together, get some feeling of what works for your problem, and bring it together. And averaging is also not always a good idea, right? Because it's often like this, that one approach works and two others don't work. So if you make the average of two things not working and one thing working, the average doesn't bring you much further. Now I just want to show you very briefly what such chemical information looks like, how we store that. And before that, I just want to, so this is basically just a very brief slide, and in the slides you can also click, so these are hyperlinks. they bring you directly to the data sources. Just to give you a little bit of an idea of what we have out there. So we have about 230,000 structures of proteins. A majority of them are bound with ligands, so it's protein-ligand complexes. The Cambridge Structural Database contains about more than 1 million compounds, but only the compounds, so not in context of the protein, where you get a 3D structure of the molecule. And this can be very valuable if someone is interested, for example, in force field development or in developing predictive methods for predicting the, what a compound will actually look like in solvent, for example, and then also protein bound. Bioactivities, again, just to give you some indicators, we have a couple of million, so a few 100 million data points on measured bioactivity data. PubChem, basically, PubChem bioassay in particular, hosts or collects data from large-scale screens. So you see a lot of high-throughput screening in there, high-throughput screening data. Campbell has a bit of a different angle. Campbell is a collection of data from the literature. So what you see here typically is smaller sets, smaller chemical series, but a very high diversity in structure and also in targets. And some of Campbell is also, and some of PubChem is also included in Campbell. So there's some overlap. I won't go too much further into detail, but you see that we have lots of data out there, but it's not data that you cannot manage. It's not like the classical amount of where you say this is now big data, we cannot handle it anymore properly, or we have challenges to handle it. And of course, lot is being done now with large language models, which allow you to investigate that data. And if you have looked at the NVIDIA stock this morning, this one is about now 12% in the negative because of this DeepSeek model, this very efficient DeepSeek model from China. Let's see how that goes. In any case, so I've always been a big fan of ChatGPT until I was in the bus today to hear, and I don't know why I even typed it, but this was like, I asked ChatGPT to predict, or to basically show me a picture of aspirin, and this is when I realized we still have some way to go. And I gave ChatGPT some further information that this is not exactly the molecule that I had in mind yet. So be careful with predictions in general. So how do we store and work with chemical information? There's actually different ways to represent a molecule. So the top one you may all know. So this is like the typical 2D representation, the structural representation, how we would basically depict caffeine. But of course, if you talk to a theoretical chemist or quantum chemist, then they would look into the electron density. If you look at people that are in haste because they're working with huge numbers of molecules, they might prefer force field representations, so something fast, like based on molecular mechanics, or graph representations. Graph representations are extremely powerful and can be are heavily used in the context of machine learning and artificial intelligence. There's molecular graphs. Basically, what the aim of that is to have the molecule represented as nodes and edges, and of course you can include properties to these edges. So nowadays, actually, one of the biggest problems that still holds back machine learning quite a bit is the problem that lots of the data is not readily machine readable. And that includes, of course, PDFs. And up here is just a link to a very interesting website from Chris Steinbeck, who developed an automated tool for the recognition of chemical structures. So if you ever have, for example, a PDF and something is drawn there and you need the smiles, you can try to use that, that approach. They are very interesting, these approaches, but there's still room for further development of these. So besides these graph representations, we have also the classical, the classical molecular descriptors. So far, there have been at least 5,000 different of those, and these can be of very different flavor. So often they are tried to be classified in these what we call like 0-dimensional, 1-dimensional, and so on descriptors. We will walk now through these one after the other, and I'll show you a little bit what the fundamentals of these descriptors are. If you go from the left to the right, we of course increase complexity, but also the information content. So this is again a slide that is just for your particularly useful for reference later. I want just to very briefly summarize it. So the most simple descriptors that you may think of when you think about small molecules or molecules in general would be something like molecular weight, number of atoms, something where you don't need to know exactly the topology of the molecule. And these are the 0D descriptors. The 1D descriptors, they are then using something like substructural information or sequential data information, and you can think of them something like substructures, for example, when you need to decide on whether something is a hydrogen bond donor or an acceptor, you need to know more than just the presence or absence of an atom, because it depends on the immediate or the proximate atom environment. The 2D descriptors, they then do another trick. They try to represent a molecule with a single number, and because they are sensitive to the size and the shape and the symmetry of a molecule, they're actually very powerful and commonly used. So even though you don't explicitly have 3-dimensional structure in there, obviously, you have some inherent properties that allow you to consider it to some extent. And the 3D descriptors, they are then of much higher complexity, where you basically aim to really understand what the 3-dimensional structure of the molecule is and to represent it. The challenge in that is, however, that the 3-dimensional structure of the molecule can very well depend on the partner it's binding to, right? So only because I can calculate the global lowest energy conformation, like I would expect it in the gas phase, does not mean that this has anything or much to do with the protein-bound ligand conformation. So this complicates things, and this means that the 3-dimensional descriptors, in a consequence, they often are not observed to outperform the 2D descriptors, but of course this depends very much on the setup and the expenses that you are in computation that you're looking at. And then you have the 4-dimensional descriptors, which can be considered something like an extension of the 3-dimensional descriptors, where you have also, where you try to consider also the dynamic behavior, so basically how the molecule evolves over time, considering multiple conformations and their transition. So that's interesting. It's basically you need to pay for a Mac to have the proper structure depicted. Sorry, this is an artifact because I'm using the presenter here. This is obviously sildenafil. So it's a very famous drug molecule. And a very simple way to describe it is basically by how many nitrogen atoms or something that I have in there. Of course, these descriptors are very quickly calculated, but the problem is they have a high degeneracy. So this means basically there are many other molecules out there that have exactly these values. It's not overly information rich. The next level of descriptors goes towards the idea of fragments, right? So basically, what has been very popular, for example, in the prediction of log P, so the lipophilicity of a compound, is to consider the compound as parts of its fragments. And each of these fragments, you can assign a certain weight or a certain contribution, which can be positive or negative. And this is actually something that has been very popular throughout many years. And it's, there's this process of replacement by more complex models, of course, more complex descriptions, also learned molecular representations that may give a more relevant and finely granular representation of the chemical or the relevant chemical information that is relevant to explain or predict a certain molecular property. And then there are the molecular fingerprints. Molecular fingerprints are incredibly well established in cheminformatics. They work surprisingly well, still surprisingly well. They're actually a very simple concept. And when they were, at the time they were developed, people did not necessarily bet on them as being really particularly good in, for example, predicting or quantifying similarity. And there are, of course, some shortcomings, but they are incredibly efficient, and one can get very, very far with them. And the basic idea is basically that you have a molecule with its topological information, so basically the connectivity of the certain element types that you see, and you convert them into a vector of features. And this vector of feature basically encodes whether a certain feature, however you define it, is present or not. For example, if this double bonded oxygen is present or not, could be one of these vector values in the vector. And over the time, this concept evolved. It started with something very simple. These are dictionary-based keys. Let me show this very briefly. Oh, Jesus. Yeah. It's not that I didn't ask for using my own laptop, but this doesn't, it doesn't work in this setup technically, sorry. So, not sure if that makes too much sense. Basically, the dictionary-based, what we call structural keys, what they are, is like, it's like just a collection of questions to which you can answer with a 0 or a 1. So basically, in this case, with the max, which were one of the earliest representations, binary representations of molecules, the idea is you have about 166 of such kind of questions where you try to characterize a molecule, represent the molecule with a bit string, basically. Is there chlorine? Yes, we can see the chlorine. Is there a ring? Not so sure, right? In this representation, we don't see that. Anyways, when you have then two molecules and you want to compare them and quantify even their similarity, this can be very simple because you have your dictionary of fragments, and accordingly, you just compare or quantify the similarity of these two bit strings. And this, from a math perspective, is a very simple and efficient approach. Then people understood that this is nice to have, but this dictionary, of course, depends always on the definition of the dictionary, right? Basically, which substructures you have to define in the dictionary. And for this reason, this concept of the fingerprints was evolved to away from a dictionary to basically containing or representing all the possible combinations of atoms that can be out there. And this is in part shown here. So this is the atom pair fingerprint, basically, which describes all the path and all the components of a small molecule. So it's a very simple molecule, where you basically have a vector of bits where you basically switch all the bits on that represent these subsets of atom pairs and their path. And what has been incredibly useful and powerful are in particular the circular fingerprints. In the circular fingerprints, you basically have an atom-centered view on your molecule, and you encode the environment around this molecule. And you can get extremely far if you consider, on one hand, the element type, and the hybridization state. So with just these two properties, you can get incredibly far, in particular in bioactivity prediction. And of course, if you have then complex molecules, you would have a bit vector of enormous size, right? Because you would need to cover all the eventualities of whichever molecular structure you will find in your molecules. And this is why the hashed fingerprints have been invented. So this hashing of fingerprints basically is something fairly simple. So it basically is the usage of a pseudo-random algorithm that assigns a couple of bits, 4 or 5 bits, to each pattern. And the only thing it makes sure is that whenever you see the same pattern, the same bits are switched on. And with this one, we can compress the representation of molecules fairly effectively. So literature suggests or indicates that you don't need more than typically about 2,000 bits, so 2048 bits, to adequately represent molecules for many, many applications. Of course, the method inherently has the risk that there are some collisions of bits, because different patterns could trigger the same bits or a subset of same bits. But this has been shown that this loss of information is actually minimal. So then let's have a look a bit more further to the 2D descriptors. So the fingerprints, depending on how you exactly use them, they are between the 1D and 2D descriptors. With the 2D descriptors, the initial idea was basically to come up with a number, with an information-rich number that basically represents the size or the molecular properties of a compound. And there are 2 types of descriptors that we distinguish. So the topostructural descriptors, which only care about basically encode basically the 2D molecular graph, and the topochemical ones that try to also consider specific chemical properties. I won't lose too much time on those 2, but I just show you one example. So this is basically the oldest of these topostructural descriptors that is linked to the branching of a molecule, and it just tries to, so here we have the molecule, thanks God, at least one molecule is visualized correctly. So, which basically, so this index tries to calculate a number, and this number is basically indicative of the branching of the molecule. And why was this of interest at that point in time? In that point in time, it was of interest because it was known that this branching is strongly correlated with the boiling points of the molecules. And so basically this number could tell you about how early or how late an alkane is likely to boil. And basically it's only a summation of all the distances, so in parentheses there are the distances between the pairs of atoms, and they are, over the summation, you get your final Wiener index. So, now, we have always been trying to, of course, get most out of, or have the representation of a molecule most accurately and most representative of its measured molecular properties. And one way to do this is, of course, three-dimensional descriptors. So the three-dimensional descriptors, what they do is they consider the three-dimensional structure of a molecule. And one typical approach to do that is to use molecular surfaces. The nice thing about molecular surfaces is that you can also project properties on them. For example, what you see here, again, our molecule sildenafil, you see there are areas in the molecule, like this alkyl moiety, which is very hydrophobic. And there are other areas, like the piperazine here. These are hydrophilic. So you can project these properties onto the surface. So the reason why I just show you this slide is maybe one thing. One reason in particular is if you visualize these molecular surfaces, there are different types of molecular surfaces to begin with, right? So there's, depending on how they are derived, so you have the van der Waals surface, you have the Connolly surface, which is the inner one here, and you have something that is called a solvent accessible surface area. And you see that they are becoming increasingly large. So the van der Waals surface area, this shows you something basically that shows you some voids artificially that, of course, the receptor is not feeling. So the protein would not feel that gap, that's artificial. But the protein is supposed to basically feel the size of the molecule of your ligand like this. And whenever you visualize, or you use a visualizer for your molecules, you will realize that there are some differences between the individual software packages and the visualization mode. So you need to be a little bit careful of what kind of structure, what kind of molecular representation you're actually looking at. Now the big challenge in using three-dimensional structures is you need to know the three-dimensional structure. You can either collect it, if you're lucky enough, from these 1 million crystal structures that are out there in the Cambridge structural database, or also, if you're even more lucky, from the protein-bound ligand conformations that are in the PDB. But most of the times, you will need to calculate it. And to calculate it, you need force fields. And force fields, in general terms, they always follow the same ideas. So you have these bonded terms, which represent the bonds, the angles, and the torsions. So the rotation around the torsional angles, and the non-bonded terms. And we are in the fortunate situation that the first two don't change so much. So the bond stretching, so the bond length, the length of a bond has such a clear minimum, and also the bond angles usually have such a clear minimum, we need to calculate them once, and they are usually maintained. So this means if a molecule approaches the receptor, we don't need to always calculate, of course, the bond length, because the bond length, the length of bonds in a molecule will not change because it's just when it binds to the receptor, right? So they are basically robust enough and defined enough that we don't need to consider them. What makes the situation difficult is actually the rotation around bonds, because many substructures of molecules are rotationally flexible, so they can basically rotate, so these rotatable bonds, and this is basically this is the primary source of flexibility that we need to consider when we work with small molecules. And it's well known and established that the molecule, the small molecule, when it binds to the receptor, what you have is something like a induced fit effect, which means that the conformation of the ligand, of your small molecule, will strongly depend on your interaction partner. And this is not just theory, but this is even seen in experimental data, right? So left, you see a molecule that everybody of you will know, adenosine monophosphate, AMP. And there are many structures of AMP in the protein data bank. And the only difference between these many structures that are here aligned is their conformation. And the differences stem not from experimental uncertainty or errors in the measurement. They stem from the fact that this molecule adopts different conformations as it binds to different proteins, or even the same protein in different conformational states. Okay? So whenever we do a three-dimensional screening, like you will see it later, for example, done in the context of the pharmacophores, it would never be efficient to work with one three-dimensional representation of the molecule. You need to always work with a whole bunch of representations, which we refer to as the conformational ensemble. So basically, the art, so basically, the science has long been to basically represent the protein ligand-bound conformations with as few as possible and efficiently as possible representative conformers, right? So basically, what you have in your database or in your screening process, you will not only screen with one representation of AMP, you will screen with a few representations, something like 10 at least, usually 25 to up to 400 representations. And here I'm only talking about conformations, by the way. Another complication would be, of course, tautomerism, protonation, and this could easily make you end up with 100-fold more molecules that you actually need to screen compared to the molecules that you are interested in, right? So basically, in other words, you may need many representations of one and the same molecule to get the right answer if you do three-dimensional virtual screening. This is just another example that maybe shows very nicely the problem behind it. These are pharmacophore keys. So this is basically the idea to represent a pharmacophore. So this means basically the interaction features of a compound as a bit string. And if you would try to consider the properties of this molecule in three-dimensional space, and this is basically one of such molecule, then you can have many different conformations, right? So basically, this aromatic moiety in other conformations can be at a very, very different place. And for this, also this fingerprint, this bit vector representation of the molecule will also change. So instead of being able to use just a single representation, you may likely need to use multiple representations of one and the same molecule. This is not necessarily a problem or something that impedes the virtual screening. It's just an extra thing to think about. And what is rather new, very hot and interesting, are the learned molecular representations, because so far we've always had the expert in the focus. So basically an expert, somebody with knowledge in chemistry, has thought about very carefully, what could be the most relevant representation of a molecule, right? The chemist would say, hydrogen bonds, hydrogen bond acceptors, aromatic aspects, log P, and so on. And there's this rather new idea to use learned molecular representation. This means basically use an algorithm, like a neural network, to pick its own representation, to decide which representations are most relevant to describe a certain task. So this means you suddenly have representations that are custom-made. They are tailored for your task. They are built in a way or generated in a way that they fulfill the task the best way. And for this can lead to higher expressiveness and information content. Basically means to just basically better represent the properties that are really relevant to describe what you're trying to describe. A big advantage can also be the scalability, so that you can handle very large data sets and also integrate different types of information. For example, two-dimensional features, three-dimensional features, and so on. And yeah, lots of research is actually going into this area. So it's not just limited to something like 2D representations, but really also sequence-based representations, so sometimes directly from SMICE representation, which is like a single line notation of molecules, all the way down to 3D geometric representations. Let me briefly show you how data is stored. And these are very old concepts. They are very, very established, and it has been like this for many, many years. But I just want to show you basically 2 or 3 examples that you really get a flavor of what we're actually looking at. This is what we call a connection table, and when you look into data files or so, they often have the extension .mol or .sd or .sdf. What that means is basically you have a file that contains the chemical information in something like a molecule block and then all your properties. And this file format is not particularly efficient, but the nice thing about it is just contains all the data. So the chemist can draw the structure, store the structure without losing anything. We can store the atoms, their connectivities. So this basically means atom number 3 is connected to atom number 4 with a double bond. So it's all very intuitive. It's basically something that can easily be read. We know the stereochemical information. Stereochemistry, please. If you start working in cheminformatics, put a yellow sticker on your screen and say, have I checked stereochemistry? Because this is like one of the biggest traps, right? Because even if you see like, I see Chiral center and somebody annotated it, what you don't know is whether they annotated it and are sure about it and whether it's the only representation, right? Because it could also be that also the other enantiomer is present, we just didn't, generate it or depict it because, for whatever reasons. So stereochemistry is something that is absolutely vital and all too often still overlooked. And then, what you can also fill, of course, is here all calculated properties, mensured properties, and so on. So it's basically a file format which allows you the lossless storage of information. Yeah, this is now sad that these really look like these structures. It's not very particularly helpful. Let me show it to you on this slide. So at some point, people said, what do we do with this file? It's nice that it has everything in there, but it's like a really bulky, and how can I carry this around and go, to other scientists and work with them? People are starting to think about single line representations of molecules, something like alongside of this. So this very simple code, it's basically, the notation is very simple to read. You can read this within 1 or 2 minutes, right? So starting to read such code within 1 or 2 minutes. This means basically the capital letters are the non-aromatic atoms, the aliphatic atoms, and the small letters are the aromatic atoms. So if we start here, so we have here the nitrogen. We never consider the hydrogens because the hydrogens can be derived, right? If I know the element type, I know the neighbors, I know the protonation state, I don't need to know the hydrogen because the hydrogen I can derive. So nitrogen, then we have a carbon atom, which is this one, then we have another carbon atom, this one, and then we have an opening of an aromatic ring. So small means aromatic, 1 means opening of the first ring, then we go through the ring. First we have an aromatic atom, a carbon, we have then a nitrogen, then we have the opening of the second ring, then we go on, then we have a branch, the parentheses indicate the branching, and then we come here, and at some point we are at this atom, this is this one here, and this atom is connected to the ring opening atom of 1 and 2. So it takes a few moments, but then this is all simple. Yeah? So you can basically describe any molecule like this. It's more or less a very efficient representation of a molecule. The only problem is, if you think about it, is why did I start down here? Who tells me to start down here? If I would start here, this whole line would look different, right? So if I would have this as a query, I would never find the molecule, even though if it's identical, but I start the representation from here. So how do I do that? There's an algorithm, or there's several algorithms out there. The beginning of that was the Morgan algorithm. And the Morgan algorithm has the idea that you assign a starting point, an entry point for this representation. And this is actually very simple. And I won't go into detail of it, but the idea is basically you look at the number of neighbors of an atom. So for example, this atom has 1 neighbor. This atom has 3 neighbors. And you collect these numbers. And after the first iteration, you go further. So you basically, you sum up again the values, the connectivity values of the neighbors. So basically, this 5 is the result of 1 plus 1 plus 3. Okay? And you continue doing that until you have the maximum number of different connectivity values. So here you have basically 11 connectivity values. 1, so this is 1, 2, 3, 4, 5, 6, 7, and so on. 11. And at some point, this number will not increase anymore. And this is the point then where you assign priorities to your molecule. And the number one priority is basically assigned to the atom with the lowest connectivity value. So then you know where the starting point is. So this is basically the starting point. There's some, some ambiguity there where additional rules are needed to basically assign the right priority to come up with the point that this is really the starting point. So this is basically the top atom to start with. And then you traverse over the molecule following the priorities. Yeah? And there are some different nuances in some of these. There are basically some different algorithms and further developments of this algorithm. So far, we looked into something that was still interpretable. So this SMILES representation is something that is really human readable and fairly quickly. Now, we look into something that basically is a single line identifier notation that aims to really collect the whole chemistry. Collecting basically whether basically at all different levels to basically encode a molecule. And this is inchy, and without going much into detail, because this looks horrific, and I'm sorry about that. Without going further into detail, so the inchy is basically an idea to basically give a unique identifier or a unique representation, single line representation to each molecule. And the nice thing is about, in particular, the latest version of this representation, that it is to the largest extent also tautomer invariant. This means suddenly you have a tool at hand where you can basically use this generator, which is freely available, and encode your molecule, and you can search your database if you find the identical string, because the identical molecule in any case will have the identical string. So you can use this to make entirely sure that you would find the molecule in the database if it is there. And then one step further to that are the smarts patterns. So people start to think about, but what if I don't want to just encode molecule by molecule? I want to encode a molecule in a more generalized way. For example, I want to have a pattern which says, have a sulfonyl moiety which can have any halogen attached, because I don't like this for this or the other reason, or I like it for this and the other reason. And this is basically smarts patterns. These smarts patterns, they build on SMILES. And they have lots of different symbols and try to be as universal as possible. And there's a really nice website from Matthias Rares' group in Hamburg, Smarts Plus. Again, if you just click on this image, you get there, which allows you to visualize these smarts patterns, because they can be very complex. Think about something like this. So you have the situation where I want to describe something like this here. I'm not going to read that, or it's too complicated. Basically, you want to have a very specific atom environment defined, and then in terms of smart pattern, this would look something like this. You see, this can be tricky to read, but this visualizer from Matthias Rares could help you in really visualizing this. So there are lots of these symbols that allow you to have really a versatile depiction of the context. And I think, or I hope you did get some flavor already of cheminformatics. Of course, within 1 hour, I'm not able to give you really all of the foundations. I hope you enjoyed it. I thank you very much for your attention. And if there are any questions, I'm, of course, happy to answer them. Thank you. Does anyone have any questions? Where's the coffee room? Just out there. Yeah, please.
I just wanted to disagree about readability of SMILES.
Yeah, I get this more often, yeah. That is right. It takes some, but it's not that we spend too much time on that, you know. Yes. And compared to the structures. Sure. Yeah. Thanks. There's the question. One. Yeah.
How do you embed angles into the graph representation?
In the graph representation, yeah, that is tricky, but you can add additional properties. Yeah, so these angles as additional properties, but I'm not so sure if there's really efficient ways out there that allow you to get much further than we are. So we actually, there's actually very interesting problems where you would need exactly that. For example, one could think about, you know, steric accessibility or metabolic stability and things like that, or chemical reactivity. And we actually tried three-dimensional representations, but nobody got really much further. For the reason that at this point you really usually get to the limitations of the data, right? So the limitations of the data set. So it's a very common phenomenon that we have, is that you get very easily very far, but to go beyond anything, you see that there's just the bottleneck more in the data than in the technology, actually. Yeah. Yeah.
Thanks for this great talk. I just want to know your opinion about data-driven descriptors, because you know, there is a lot of discussions now about data-driven descriptors, and if they really outperform like classical ones. What's your opinion based on your experience?
My personal experience is that we have always tried them out. It's like just one set, right? We do all the fish scan, we do all the fingerprints and the new learned representations, and so far we've found them never to outperform or have a significant advantage. But I'm hopeful that we will find that at some point. It is interesting conceptually, but so far I think the data is the limit, at least from the problems that we are working on. We are usually we have very small data sets, you know, that we are working on. Yeah. Yeah, thanks very much. So I think we can have the coffee break now, right? Yeah. Thanks, everyone.
Okay. So good afternoon. Nice to see the most reliable participants of this conference. At least half left, but at least the teachers are here at the school. It's already a good sign. Okay, so I will speak about chemical space. The batteries are coming. Perfect. Yes, something, or at least this. Okay, but this is already not bad. Okay. Good. Okay, before starting to speak about chemography, let me discuss what is chemical space. What does it mean, this term? So, is this ensemble of molecules which Professor Kirchmair actually mentioned in his talk, or this is some sort of mathematical objects. And by the fact, in the literature, both these views are discussed. A famous paper by Chris Lipinski and Andrew Hopkins, they spoke about chemical space as an analogous to cosmological universe. And chemical compounds were associated with stars and planets. And we decided to reconsider this. And from a mathematical point of view, space is an ensemble of objects with relations between them. And very recent paper by Jean-Louis Raymond also he formulated chemical space as mathematical and usual, as I said, usually high-dimensional space, where distances represent similarities, etc., etc. So actually, we have two points of view, and both points of view are used in the current literature. So I will speak about space as a mathematical object, and which must identify the objects in this space and relations between them. So in cheminformatics, the main objects are two types of structural representations, either as molecular graphs or like as a vectors. And so many different concepts are considered in a graph-based chemical space, like scaffold analysis, scaffold tree, molecular match pairs, activity cliffs, and chemical space networks, for example, are MMP-based. And mostly I will speak about vector-based chemical space, which allows to analyze and visualize the data in n-dimensional space of molecular descriptors. And this mathematical object nowadays, now I will speak about ensemble of molecules, this mathematical object may describe, accommodate a huge amount of data. Professor Kirchmair already mentioned the numbers. So the databases of available recorded compounds, they contain some hundreds of millions, and 10 to 6, 10 to 8. 10 to 10, these are billions, tens of billions. This is a real space of Yanomir, for example. And 10 to 20, 10 to 26, these are proprietary databases of big pharma companies. And also this star, this actually estimations of Pavel Polishchuk based on the evolution. This actually mathematical estimation based on extrapolation of the all possible graphs which could be enumerated for a certain number of molecules. And this is the lowest bound of, not of any molecules, but of drug-like molecules. So this is a minimum, but still it is a very big number. Why do people need so large databases? Because they expect that the larger database, larger library, increases the chance to retrieve hits in virtual screening. Let's say this is very questionable because screening success depends not only on the size of the database, but also on the chemical diversity of compounds. So that's why we need a tool which allows to describe this huge volume of data, accounting for chemical diversity. And today we'll speak about chemography. These are magic maps, which allows to do many different things. So, and from the practical point of view, may help to prepare on-demand library. And in combination with the modern AI tools, may help to design novel molecular entities. So the idea is very easy with these maps. We speak about vectorial chemical spaces. So first we calculate descriptors or learn representations, whatever, then each structure is an object in n-dimensional space, which we reduce to 2 using dimensionality reduction techniques. They exist dozens, if not hundreds, different dimensionality reduction techniques. So these are, this is example, shows application of some of them for the same data set from the due data database. And for some reasons, we selected one of them. Further, I will show one of them, which is called GTM, Generative Topographic Mapping, which was developed by Christoph Bishop as a probabilistic extension of self-organizing maps. So before telling you about GTM, yeah, we have been using GTM for already more than 10 years in different cheminformatics contexts, in different scenarios. And today I will show you some of them, case studies, which to my opinion may confirm utility of this approach to all these typical cheminformatics tasks. Okay, before speaking about GTM, I would like just to speak about descriptors. So we use our in-house descriptors, which are called EZIDA. EZIDA, this is a project running in the lab in silico design and data analysis, and which we use as descriptor molecular subgraphs, substructures, which may differ by their size, by their topology, either paths or atom-centered fragments, and content, so we can use either atomic elements for the nodes or, for example, pharmacophoric labels. So in such a way, for one data set, we can generate several hundreds different pools of descriptors, and then we can select one or several the best according to efficiency in particular machine learning task. Okay, now coming back to GTM, very briefly about algorithm. So the algorithm inserts in n-dimensional space the flexible sheet. Christoph Bishop, the father of GTM, called it a rubber sheet with the internal dimensionality 2, which covers the most dense areas of the data points. And there is a rectangular grid in nodes of which the algorithm fix Gaussian functions, and this ensemble of Gaussian functions is supposed to characterize the data distribution. Okay, and since each data point is characterized by several Gaussian functions, so the projection is fuzzy. It means that it is not just a data point and x and y. It looks like a ink drop spread over the paper sheet. And then we accumulate these drops, and finally we get such called density landscape. It means it is accumulation of all data sets, which by the fact can be characterized by a simple vector, which is built on the density values in the nodes of this grid. Okay, so one database, whatever the number of compounds is inside, is characterized as one vector. And in such a way, we can easily compare two databases of any size. Moreover, we can consider different, let's say, version of this same map. Using the color code, we can introduce some information about activity, either binary, active, inactive, So in here, this is map in the middle is, we call this a class landscape, in which zones colored by red are populated exclusively by active compounds, in blue inactives, and yellow and green by both. And another landscape we call activity landscape, in which we use just the numbers. For example, on the right, this is the molecular weight landscape, in which red zones are occupied by heavy molecules and blue zones by light molecules. And certainly each of these landscapes can be encoded by a vector. So the density landscape characterizes only by structures of molecules, and class and activity landscape, both structures and activities. So we interestingly that these landscapes play a role of machine learning model when we project a new molecule on the map. So activity landscape predicts activity value. Class landscape predicts a category active, inactive. And if the molecule drops in the empty zone or low density zone, it is considered to be out of the model applicability domain. So this means that GTM actually allows to identify its own applicability domain. I will not speak too much about application of GTM as machine learning method. Certainly it is a bit less well-performing compared to classical approaches like random forest or SVM, although in many cases this performance in cross-validations is pretty similar. So let's consider a different scenario. First, classical, how GTM can be used for data visualization and analysis for particular database. So this is a Campbell database, about 2 million compounds. And if you look closely to the clusters, so you see here some dense areas colored in red and yellow, green. So they are mostly populated by some particular chemotypes. So this map can distinguish different chemotypes. This is already good news. And moreover, if we, since Campbell database actually is annotated with different activities, we can build complementary class landscapes for subsets related to particular biological targets. So altogether, for Campbell, we built more than 700 such landscapes, which means that for any new compound projected on the map, one, first of all, can identify the most similar compounds from Campbell and predict its pharmacological profile for several hundred biological activities. In fact, for particular databases, GTM is, well, if the database is not too big, the this method can be compared with other popular methods like UMAP, TSNE, etc. But with the most powerful GTM is when you want to compare different data collections. So let's, on this scheme, consider library 1, library 2. And if we superpose them on one map, we can identify zones which do not overlap, and these zones contain unique chemotypes for one or another database. And this is much more informative compared to classical databases analysis based on this one-dimensional data distribution like histograms. This example shows distribution of log P, hydrophobicity for two libraries. Both have the maximum at log P equal to 4, and the conclusion which you can draw from this two histograms that the two databases are very similar, which is not the case. If you consider density landscapes, they are very different. So conclusion is that certainly two-dimensional distribution gives you much richer information about the two databases. Okay, now with respect to comparison. So let's consider case study when we overlap Zinc database containing commercially available compounds with the Chem database containing biologically relevant chemotypes, and the idea was to identify unique chemotypes for both, which may help compound suppliers to think about enrichment of their stocks, and people doing screening to take some new chemotypes in their screening collections. So altogether, we considered about 1 billion compounds, but this example contains only fragment based on lead-like compounds, and this subset contains only 3.6 millions. Here, zinc is in red, chem is in black, but on this map, you don't see neither red nor black. So what does it mean? It means that the map resolution is not sufficient to separate the two classes, the two databases. It looks like you have a world map, and from world map you want to identify holomodes and even this building in holomodes. Not possible because of resolution. So what usually we do in geography, we prepare another map. You know, map of Europe, then map of Czech Republic, then map of holomodes, and then quarters of holomodes, etc. So we make some zoom. So what does it mean? So we select a zone, in this particular case, zone 1, and we build, this is not optical zoom, we build another GTM. And this is not sufficient because still we have a lot of green zones, and we continue until the two databases are well separated. And when they are separated, from this level 2 map, we can extract unique compounds, and for which we can make scaffold analysis or extract maximum common substructures, etc., etc. So in such a way, we built for this 1 billion system, we built more than 40,000 hierarchically related maps, and in which we identified more than 100,000 chemotypes never been biologically tested, and more than 20,000 chemotypes missing in commercial space. So, and certainly we built a tool, web-based tool, which allows to navigate. So we call this ChemSpace Atlas. Okay, another very practical task, which is pretty often, this common task for pharma companies, is reshaping proprietary collection. So I had a PhD student, Arkady Lin, who was shared between my laboratory and Boehringer Ingelheim. And colleagues asked him to use cartography to select from Sigma Aldrich catalog some new compounds. They had a limited budget, roughly for 1,000 and half compounds. And what did he do? He overlapped on one map the proprietary collection 2 million compounds from Boehringer with 8 million from catalog, and from which, well, he made zoom, etc. And then finally he used a certain number of filters, Lipinski. Then he predicted activity with GTM landscapes, and then paints, etc. And finally, he arrived in a reasonable amount, 1.2 thousand compounds, which then were transmitted to the management department. I don't know if they really purchased these compounds or not, but at least the colleagues from the screening facilities in Berka were pretty happy with this selection. Okay, now last case for pairwise comparison. So recently, Yanamin, ChemSpace, they announced the preparation of such called freedom space, which, according to them, was complementary to real space, and they couldn't give good confirmation of this. So we, in collaboration with them, we made this cartography analysis, and indeed, on the map, we see very good separation of the both databases, freedom in black and real in red. And then we certainly extracted chemotypes unique for both, etc. And then we demonstrated that this new space, freedom space, is enriched with drug-like compounds, etc. So this is a good demonstration as a practical usage of cartography to analyze the database overlap. And then let's go further. Let's suggest that we have many libraries. So we have one reference library and many we will call external libraries. And we want to, let's say, to identify among those external libraries the one which is the most similar to the reference one. And we will consider a case study related to the situation when we take Campbell as a reference library and DNA-encoded libraries as the external libraries. So that means that these external libraries, they could be considered as the best candidates for primary screening when we don't have a particular target, but we need the most diverse library for screening. Okay, so the idea is to calculate similarities, pairwise similarities between reference and external, and then rank them according to the similarity value. Okay, let's consider how does it work for DELs. Just to remind you, the DNA-encoded library, or DEL, represents combinatorial collection of small molecules covalently attached to a short DNA tag. So, so if you have, let's say, we have this combinatorial building blocks linked according to reaction rules, and each building block comes with its DNA tag. DNA-encoded library is a very interesting alternative to conventional screening. In screening, you prepare the, yeah, it doesn't work, the point. Okay, on the left, so you see the separate wells, and in each well, you have individual compounds, and you can actually modify this collection by cherry picking, replace one compound with another one. This is not the case with DNA-encoded libraries. With DELs, you synthesize the DELs in the single test tube, and you put a protein on the solid support, and then you take this protein out with some molecules which are bounded to this protein, and then you identify these binders according to DNA tag. You make some amplification, and you identify the molecule. So this is very simple as concept, but in practice, it is pretty expensive. So in fact, if you have n building blocks, a library of building blocks, and m reaction rules, you can generate thousands of different libraries. Not compounds. You generate billions of compounds and thousands of libraries. And experiments with one such library typically is, you know, it costs quite a lot. You know, according to some estimation, it's roughly 120, 180,000 euro. And duration, so I spoke with the Astra people, roughly 4 months. So it looks like very easy to do, but in fact it's not so easy and expensive. So, and the goal is to select an optimal DEL for particular task. Okay, the idea is very easy, you know, so we can generate many DELs, and then since GTM allows to encode each library by a vector, we can simply compare the vectors using any metric, for example, Tanimoto. So this is a case study. So we generated more than about 2,500 DELs, raising from 1 million to 1 billion. Then we selected representative subsets, 1 million each per DEL. So altogether, this corresponds to 2.5 billion molecules. And then we performed pairwise comparison with Campbell, and then ranked these similarities, and then selected the highly scored DELs. So this is the result. So the best DEL, you know, already provided with a more than 50% of coverage of chemotypes with respect to Campbell. So this is a related overlap. DEL is red and Campbell is black. Then we, let's say, added another, you know, well-ranked DELs. Already 3 DELs is about 80%. Then 5 DELs more than 80%. And then what happens when we overlap all DELs, we discovered gray area, which is not covered at all, because this area is populated by natural product-like compounds, which are orthogonal to DEL-related chemistry. So in such a way, we can select DELs which the best fit the Campbell, and we did this pretty quickly, despite the fact that each collection contains millions of compounds. So now we can go further. If each library can be represented as a vector, we can construct such called chemical library space, in which each object is a library, and build another GTM for this new, let's say, n-dimensional space. So we call this meta GTM. And each data point is individual library. Here, this is an example for these 2,500 DELs. We have here the blue star is the reference library. The red star is the closest DEL, and gray star is the most remote DEL. And as you may see here, the landscape, density landscape of the closest DEL, let's say, is not the same as reference, but at least it fits the best than the most remote DEL. And moreover, we can actually make more deep analysis, just look at which reactions were used to synthesize DELs. The most, as this scheme shows, this map shows that the most of DELs are covered by coupling-based libraries, and very few actually are built with a heterocyclization-based reactions. Okay, now what, anyway, so in previous examples, we selected 1 million from the entire DEL, which may encompass 1 billion and more. What happens if we want to consider the entire DEL up to 1 billion and more? So the bottleneck here is when we make this projection, so from building blocks, we need to enumerate, standardize, calculate descriptors, and then project. And for one CPU, processing of 1 million compounds, with more than fingerprints in this case, actually takes about 12 hours. So 1 billion, it means forever, more than 1 year. So what to do? We decided to build neural network, which should be called CoLIN, Combinatorial Library Neural Network, which allows to build GTM directly from the structural building blocks and reactions, okay? And it completely skips enumeration step. And 1 million actually takes only 1 minute for one CPU. It's significant acceleration. So I would like to show you this architecture. It is, actually the paper is in press, will be published soon, but it is available in Chemarchive. Just some results. On the right, you see the similarity of two databases, two maps. One map is built for the density landscape of the library of 90 million structures, and the right map is the data density predicted with the neural network. And then the horizontal X shows the size of the data set. And you may see that training set containing only 25,000 structures is already sufficient to explain huge library of 90 million compounds. Yes. So this is interesting way to, let's say, work with ultra large chemical data. So just, I would like to notice that Sergei Sosnin is here in the audience, so I would like to mention his work published, let's say 8 or 10 even years ago, something like that. 10 years, yes. His similar approach for parametric TSNE, he used, he built neural network to identify projections in the TSNE map. Okay, now quickly I would like to turn to our biological structures, because according to votes organized by Professor Kirchmair, some of you prefer biological molecules instead of small molecules. Okay, what about biological molecules? Okay, we will consider GTM in the context of drug resistance for two objects, for virus. Certainly, this is HIV virus, and one bacteria, Staphylococcus aureus, nice bacteria, basically. Okay. I would like to tell you more than here about the danger of this virus. What is important and very unfortunate, this virus mutate. What means mutate? Mutate some proteins, you know, especially these two protease and reverse transcriptase. Let's say this, on the right, you have structures of two drugs discovered pretty effective against these proteins. But if you, let's say, if they mutate, these drugs are not efficient. And the bad thing that this mutation may happen pretty far from the binding site. So you actually, 3D methods are not, let's say, reliable, because this mutation may happen somewhere. And this means that if the medical doctor prescribes the drug, it could be efficient or not efficient, you know. So that's why we can suggest computational approach, which, according to the composition of the strain, allows to predict which drug can be advised to treat particular patient. So this is a way for personal medicine. So, okay, how did we proceed? So we selected from Stanford HIV database more than 4,000 proteases and reverse transcriptase amino acid sequences and associated profiles with respect to altogether 14 drugs, existing drugs, 6 protease and 8 reverse transcriptase inhibitors. So this looks like this. So for it doesn't work. Okay, you know. There's a real problem with batteries in this country, to notice. So for each column, this is resistant or susceptible with respect to particular drug. So as descriptors, we used such called K-mers. These are very typical descriptors for biological sequences. These are counts of the subsequences of K. In our case, K was equal to 3. And then, let's consider this protease, which is not too big. This is a homodimer and about 100 amino acids per monomer. And then we built a resistance landscape for two drugs, nalfinavir and darunavir. And you see there is a very clear separation between resistant and susceptible. And if we consider the two zones just in frames, so you can see that some zones which are resistant, these are sequences which resistance to nalfinavir, but not resistance to darunavir. And then moreover, we can also associate with these two landscapes another landscape, this landscape which shows mutation patterns. And these patterns of particular mutations. And then in such a way, we can, in this zone, associate and, let's say, interpret resistance to nalfinavir to these particular mutations. So this is just an observation, which then in the literature we discovered that actually it is confirmed by experimental studies. So in such a way, these maps, certainly we can also build SVM or random forest, whatever, can be used for personalized treatment, not only to, let's say, to advise the patient this or that particular drug, but also, let's say, to associate resistance with some particular mutations. Okay, very quickly, another example with antimicrobial resistance, Staphylococcus aureus. Look, it's very, very bad pathogen because actually, if you have it, the patient which has methicillin-resistant Staphylococcus, actually only 1/3 has a chance to survive. So, okay. Now, how did we build a resistance landscape? So we selected the data from 5,700 something from Patrick database. Unlike proteins, these are pretty long sequences of nucleotides, 2.8 million base, and then as a consequence, you have several tens of millions of K-mers, of descriptors. So this is a huge computational problems. So how did we actually manage to solve it? So we used kernel PCA to reduce, let's say, millions descriptors to only a few hundred descriptors, which then used as an input to GTM, random forest, SVM, etc. And then we also identified, by comparison, this resistance map of different antibiotics, we could actually associate the drug resistance with the particular the expression of particular proteins like PBP. This is the penicillin protein binders to A, or beta-lactamase, which beta-lactamase, according to this comparison, actually is responsible for penicillin resistance, which actually also confirmed by independent experimental measurements. Okay, now let's speak about AI tools. Now everybody does AI. It's very, even my iPhone, actually. And I will show how actually GTM and cartography in general can be helpful to generate new molecules, reactions, and peptides. Okay, the idea is very simple. So we have this is a learned representation. This is a case of autoencoder, stem autoencoder, but whatever we can use transformer when we build vector from text or graph. And this vector, actually, in this vector, each molecule is an object in this new n-dimensional space. So the dimensionality, this is actually user-defined parameter. And then for given seed vector, we can associate the Gaussian noise, and then we can sample from this Gaussian noise, and then decode, if we are lucky, so this new generated vectors represents valid smiles or valid graphs. Okay, so in fact, GTM helps us to identify a seed vector from which valid structures possessing a given activity can be generated. So we did, let's say this is an example, so if for ibuprofen as a seed, it generates its analogs. And then, well, related to GTM, we select the zone. And if we select a zone, we can, let's say, believe that most of molecules of structure generated from these zones will also be potent. This red color corresponds to high activity. Okay, so we published a paper 6 years ago, and we discovered that indeed generated structure are enriched with new scaffolds, and let's say we did not perform experiments, but according to docking, the structures were pretty efficient. And you know, this is a very fast way to guide artificial intelligence to the area to be sampled. Certainly, maybe it is a bit less efficient than reinforcement learning, but it's a very simple and straightforward procedure. What about reactions? You know, look, the gold medal on the right. You know, according to statistics, each new type of chemical reactions actually is awarded by the Nobel Prize. And our motivation was, if we ask a computer to generate us the new types of reactions, maybe we can share with the artificial intelligence new Nobel Prize. Okay, so yeah, the problem with chemical reactions is their complexity, because a reaction equation contains several molecular graphs of two types, reactants and products, and then the first one, and how to detect novelties and reaction feasibility. You know, well, novelties for individual molecules is scaffold-based. What about reactions? So in order to tackle this problem, we used another concept which allows to reduce complexity of reaction equations. We call this condensed graph of reaction. And the idea is to condense all molecular graphs involving a reaction equation in one sole molecular graph. So in such a way, instead of these two graphs, three graphs, we can, on the left, we can represent them as a one single molecular graph, which is characterized by both conventional chemical bonds, single, double, aromatic, and such called dynamic bonds, formed or broken or transformed. Here, green line corresponds to formed bond, and pink lines corresponds to broken bonds. Okay, so and this is new one single molecular graph, so we can consider this as a pseudo-molecule for which, by the fact, we can build use Smiles notation, and this Smiles notation is almost twice shorter than conventional reaction Smiles, because each actually character, each atom is only mentioned only once in this. And this is very good for Smiles-based AI tools. So we did this, we trained the autoencoder on USP2 database, about 3 million reactions. And finally, then we colored this map with the reactions corresponding to Suzuki coupling. And finally, we sampled from the areas of Suzuki coupling, and we generated 13 Suzuki-like reactions. And then 5 of them were found in SafeFinder, and SafeFinder, which is much larger database than USP2. So let's say, unfortunately, these are not this is actually a bit exotic coupling or exotic living groups, if there are some chemists among you. No Nobel Prize for us, but we were happy just that it was implicitly validated by comparison. And then a very recent project related to a generation of new peptides, which ended up with the experimental validation by the fact. So we, for methicillin-resistant staphylococcus, I told already, so we built Wasserstein autoencoder, from which we sampled and then selected some peptides and which were tested. Just to tell you that these were short peptides up to 14 amino acids, only natural and only linear, okay? And then we used, in the data set, we used not only unlabeled peptides, but also 680 peptides for which we had experimentally obtained labels against this methicillin-resistant staphylococcus. So finally, from this sampling, we ended up with 8 peptides. Why 8? Because actually it was experimental people asked us to give them a very small sample. We provided with 10, and then they managed to synthesize only 8. And all 8 were active against biofilm. 5 out of 8 active against MRSA apoptonic cells compared to control, and control was pretty serious. So this is in bioarchive and is submitted already for publication. Yeah, just to end up with this story. So for the moment, we sampled from the latent space of autoencoder. But look, this autoencoder latent space, not necessarily the best descriptors for machine learning. It could be that classical molecular descriptors for the given QSAR task represent better descriptors. So they provide with better models. But on the other hand, classical molecular descriptors, they don't allow us, this space don't allow us to generate new structures. So what did we do? We developed a neural network architecture able to generate structure using any descriptor space. And just to give you an idea, that for training this, we built attention-based conditional variation autoencoder. As an input, we used both smiles and descriptors. And when the model was trained, we submitted only descriptors. In such a way, we managed to solve inverse QSAR problem when we have optimal descriptors and the model, related model, and we can generate structure which corresponds to particular descriptor vector. Okay, I have still very, very few slides, almost finished. So nowadays, people use massively UMAP, PCA, TSNE, and you will have a tutorial related to this application of this nice, popular data visualization techniques. Recently, we performed a benchmark of all these techniques, and including GTM, using more than 100 subsets from Campbell, 3 types of fingerprints, and we made comparative analysis of neighborhood preservation according to 18 metrics. So the paper is published. I would like to give you details Otherwise, actually, it will need me to take one hour more. Conclusions here, you know, with respect to neighborhood preservation, TSN EU map and GTM, actually they are more or less at the same level. But with respect to out of sample, what does it mean? When we project new compound on the map, which was not used for training, we have clear preference of GTM. Yes. Just to summarize, why GTM? So, conclusion of all my talk, GTM considers data probability distribution that makes it suitable for big data analysis, first. Manifold can be built on small representative set of data and then can accommodate a lot of new data. This is very important. You don't need to build this map on millions. You take very small portion. Then landscapes, GTM landscape can be used in regression or classification tasks. Data collection can be encoded by a vector, which enables fast chemical libraries comparison. Also important. This is actually unique niche for GTM, I believe. And then coupling of GTM with AI technologies facilitates de novo design. So I would like then, in the end, to show you my teams contributing to this works in Strasbourg, one in Strasbourg, and another small in Hokkaido. Numerous collaboration with academic and industrial partners. And the very, very last slide. Maybe you know this personality, this brave soldier Švejk, and this actually, he missed his way, you know, to České Budějovice, probably because he didn't use GTM. Thank you. Questions?
I was curious about the, you point out when you compare GTM with other, let's say, dimension reduction techniques, there is much better neighborhood preservation for out-of-domain data. So how come it's like that? What's the, because to some extent it's learned, which is why in case of Disney and UMAP this fails a little bit when you have out-of-domain data in there. So how come in case of GTM this works a little bit better? Is there some like...
No, it works because if, let's say, GTM model the data distribution. If the new structure belongs to the same data distribution, let's say, because there is a particular parameter which controls this, it's called log likelihood. If it belongs to this distribution, actually it is already described by the manifold. Manifold is a model, GTM model, in fact. That's why.
I see. Thank you.
I just wanted to comment that Švejk did not take the direct route because he wanted to avoid the war.
What? Švejk?
Yeah.
That's true. That's true. That's true. Yeah, I advise everybody, you know, to read this fantastic. This is one of my favorite books of Jaroslav Gašek. It's still very, very in actuality, you know. Okay, Sergey.
Thanks for this wonderful talk. Yeah, and my question is, you mentioned the geographical maps, like projection of chemical space. We can look at this as geographical maps. But geographical maps, they have one peculiarity. They have standardized coordinates. So we just, that's by our choice, that zero meridian goes through London, for example, and everyone knows this. But for GTM, for any kind of projections, we don't have such standardization. What do you think? Is it worth doing like if we build a GTM map, for example, on all PubChem compounds, and say for chemists, like we have in this region, we have steroids, in this region, we have benzodiazepines, in this region, and standardize by coordinate level?
Look, you know, so we consider some references in the world map, because we are still actually linked to this planet. Let me consider more planets. What about reference? We don't know. You know, the chemistry is large. I spoken about ultra-large spaces, you know, containing billions and billions, you know, so we can actually consider different subsets, different planets, and different stars, how to identify the reference, you know. Even let's say, if you take, okay, if you want to build a map of, let's say, currently known compounds, currently synthesized compounds, certainly we can make an effort. But what happens if, let's say, tomorrow we'll discover new class of molecules? So we will need to reconstruct GTM, because these new structures could be out of obligability domain, and they could not be well described by the manifold. You know, so it means that this planet is already well studied, it is established. We can hardly believe that the continent will move for at least for our lifetime, you know. That's why we can easily identify the reference and play with. I don't believe that it's worth to do. And then, you know, the question of descriptors. The map,
Of course.
certainly, you know, varies as soon as you change descriptors. So I wouldn't do this, and I wouldn't establish the word chemical map using GTM or UMAP or whatever, you know. So look, I like GTM, but we should not forget that GTM is one of many different methods. And there exist no magic methods. No magic methods, including GTM, which explain and characterizes and analyze everything. This is one useful tool. Like, you know, in dentist, when you go to your dentist, it has many tools. And he or she uses the tool as a function of your problem. If I use only GTM, I would not be able to, let's say, to model too much things. So GTM has its area of application. And some of these areas of application is unique, but we need other tools in combination with this, for sure. Thank you.
Well, thank you for the first day of the workshop. And now we will have the poster session, and there is coffee as well.
Thank you for sending it. So is MSD already paying for GTM license to Strasbourg?
No, but we have a project that involves the Bells library.
Yeah, of course. Actually, MSD's Dell compound was one of my favorite big small molecules of the last years. It's like respect for that one.
I was involved in the disciplinary papers, but I let that go. So we're building a bigger platform that should include.
Something a bit.
Yeah. I forgot how I put So now it has to be. Yeah.
Oh, yeah.
Somewhere. Yeah. Yeah. Yeah, I really look like Martin Schiffman.
Yeah, the Dell comparison idea, actually following Dell's, it's a pretty conceptually cool idea, whether you do it with GTM or otherwise. Although, to be honest, Dell... No, it was just a one-sentence remark I was going to make. Actually, like Dell readouts, compound per count Dell readouts, this is like horrible data to deal with. Actually...
Yes, that was the pipeline. I mean, that was the biggest part of the pipeline to somehow automatize it.
Because this is so super noisy, because a lot of it is like synthetic, let's say, like 99%. Also, like, let's say, if you have a small molecule that binds in a pocket, and the small molecule is conjugated to DNA, it means that the part where the DNA is hanging on it, it needs to have place. So, actually, you reject a large amount of compounds.
Yeah, unless they're on the surface.
I'm not sure if it's worth the 150,000 euros. Yeah, this is like actually this. Maybe you remember this Berka challenge on Kaggle. It was like some Dell data with like 300 million compounds in there, and they were like, we're gonna revolutionize drug discovery in this biggest bioactivity data set ever. And like, you know, thousands of people participated in this challenge. Predictions were, of course, horrible. And then later I read a biographic article about the guys who made this data set, and it was like some guy who doesn't know about biology, who bought some machine, and who did those experiments in his basement. So it's like very obvious that data is like complete trash.
У мене доповідь була в останній день. Весь час, що то все краще.
Brutally criticize some posters now.
I have 5 or 6.
Do that.
So is there going to be poster?
Can we leave our stuff here still, or should we take it?
Okay.
Well, there's probably no one coming.
Okay,
You can
I
leave
can
it
put it.
here,
I
but-
can just put it on a chair. It's fine.
There's nothing more.
One possible solution for you is to leave it in the circle.
Oh yeah.
Because
In the where this focus pendulum is.
so in the center of the.
So no one will dare to go because they're afraid they will get hit by the cannonball.